Hello everyone. Welcome to this tutorial on lower motor neuronal control. This is really the first of a set of new topics that we will consider, where the primary focus will be on understanding the way the brain controls voluntary movement. And we begin by considering the lowest level of this set of neural elements that run between the cerebral cortex and the muscle tissue that actually does the movement of the body. So this tutorial will unfold in 4 parts. This first part, I'd like to give you a bit of an overview of the somatic motor system and define for you and talk just a little bit about motor units. For this first part of our tutorial, I have 2 core concepts in the field of neuroscience in mind. Again, the complexity of the brain is always before us, and so I'd like to not miss an opportunity to remind you that the brain is the body's most complex organ. And again, most of what we'll be talking about today pertains to the organization and function of circuits that are genetically determined, and they form the foundation for the nervous system's ability to execute voluntary movement. So I've got some learning objectives that pertain to this first part of the tutorial. I'd like for you to be able to discuss the general somatotopic organization of the motor neurons that are found in the ventral horn of the spinal cord. And I'd like for you to be able to characterize the motor unit and discuss different types of motor units. Okay, so let's get into this by talking in broad terms about the organization of the human central nervous system. Now, you've seen this figure before, and I used it to introduce the concepts of central nervous system, peripheral nervous system, and the general phenomenon that is so critical for understanding human behavior, and that is sensory motor integration. Well, now that we have completed our survey of the sensory systems that take information from the internal and external environment, process that information along the way between the spinal cord, the brainstem, the diencephalon, and then finally up to the level of the cerebral cortex, then that information becomes synthesized and integrated in circuitry that is distributed across the lobes of the cerebral hemispheres and runs loops into subcortical structures such as the basal ganglia and even back down through the thalamus and through emotional centers, including structures like the amygdala, and memory centers, structures like the hippocampus and the parahippocampal gyrus. Well, ultimately, this process of sensory motor integration gives rise to behavior. And it's to the behavioral response that we now turn. And so what we're going to be talking about are components of our motor systems. And as this chart is attempting to make clear, there are parallel pathways that allow our motor system to produce behavior. There's a channel that we'll have a tutorial about in just a little while, talking about the autonomic nervous system, or what we sometimes call the visceral motor system. But for the next couple of sessions, I really want to focus in on what we'll call the somatic motor system. This is usually what we think about when we consider human movement. We're thinking about the activation of musculoskeletal effector systems and the production of physical movement of our frame. And so this involves the activities of central nervous system stations that send signals out through motor nerves to striated or to skeletal muscles, and that actually produces the movement. So we're going to work our way into this motor system one step at a time. And this figure now really lays out a framework for understanding this somatic motor system. And let me highlight a few of the key features here. First of all, we'll work our way from below and then up through this motor system. And we begin with this idea that the skeletal muscles are the effector systems. And so if we want to understand the control of the skeletal muscles, we need to consider the neurons within the central nervous system that innervate those skeletal muscles and cause their contraction. These we call lower motor neurons. And so these lower motor neurons exist within a lower motor neural circuitry that is found at every segment of the spinal cord, and for the cranial nerve motor nuclei also in the brainstem, in association with our cranial nerve motor nuclei. Now, this circuitry that we find receives sensory inputs, and so one dimension of activity that we will explore today is this idea of local segmental circuits that perform reflexive adjustments of movement in response to specific sources of sensory information. So the local circuitry of the spinal cord and brainstem mediates reflexes. There are also circuits that span the segments of the spinal cord, and in some instances span levels of the brainstem, where we find different kinds of cranial nerve motor nuclei. These kinds of circuits we call intersegmental circuits because they are required to coordinate the activities across the longitudinal length of the central nervous system where we find our lower motor neurons. One example might be swimming, or perhaps swaying our arms while we are ambulating on our 2 feet. So obviously in both of those activities there's coordination between the movements of our arms mediated via the cervical enlargement of the spinal cord and the movements of our legs, which are coordinated at the level of the lumbosacral outflow from the spinal cord. Now let's move up the nervous system just a bit and consider how is the outflow from these circuits that govern the lower motor neurons controlled? Well, they're controlled by a set of neurons that we call upper motor neurons. And these upper motor neurons exist in the motor cortex and in the brainstem. Now, I want you to notice the upper motor neurons themselves do not innervate skeletal muscle. Rather, they provide descending input that are governing the way the local circuits work to organize the outflow of the lower motor neurons. And in some very special instances, there may be direct projections from upper motor neurons to the alpha motor neurons that actually do supply innervation to skeletal muscle. But notice for the most part, this arrow is a lot larger than the one providing direct input to the alpha motor neurons, which is to imply that the way our motor system is organized is that the upper motor neurons largely coordinate the local circuits that then do the job of organizing the output to skeletal muscle. Now, when we're thinking of movement, we are often thinking about voluntary movement that we consider and that we actually express through active will. When we are thinking about voluntary movement, you should be thinking about activities that are planned, that are coordinated, that are initiated at the level of the motor cortex. The motor cortex is the posterior part of the frontal lobe where our executive functions are integrating sensory and emotion and mnemonic signals and devising plans for the production of behavior. And the actual final signal to produce that behavior is a complicated process that we'll talk about in subsequent tutorials. For today, I want you to understand that these are the kinds of activities performed by the motor cortex as we prepare to execute a voluntary behavior. And then ultimately, of course, that information is relayed down to our lower motor neurons. Now, in addition to the motor cortex, there are also upper motor neurons in the brainstem. And these motor neurons are involved in setting the stage for the execution of voluntary movement. Now, much of the voluntary movement that we make, we can think of it as the expression of skill. Consider, for example, typing on a keyboard or using a pen or a pencil or some other writing implement, perhaps performing dance or music with a musical instrument. These are all aspects of skill, and primarily skill is expressed through the activities of our hands, our feet, and the lower oral regions of our face. Well, in order to be skilled in these body parts, we need to have proper adjustments of posture, and we need to have the proper amount of tone present within the muscle systems that actually express that skill. And this is largely where these brainstem centers come in. So while I don't particularly think of these brainstem centers as expressing voluntary behavior, I do think of them as being essential for the expression of the skilled voluntary motor acts that we normally think about when we consider human movement. So from these brainstem centers, for example, we have the coordination of posture, as well as the organization of basic movements that are essential for sustaining goal-directed behavior, such as locomotion and other aspects of sort of rhythmical, highly stereotypical activities, including things that we do with our cranial structures, like chewing. Well, there's another aspect of upper motor neuronal control that we'll talk about a few tutorials down the line, and this is perhaps somewhat at the interface between what we might call voluntary and involuntary behavior. And what I'm thinking of is the expression of emotional behavior. Emotional expression reflects the coordination of both somatic motor and visceral motor activities. And this is not so much a function of the posterior part of our frontal lobe. Rather, it's a function of those parts of the brain that tend to be present mostly on the ventral and the medial aspects of the forebrain, involving structures like the orbital aspect of the frontal lobe or the medial midline aspect of the frontal lobe. And these structures are interconnected with structures in the temporal lobe, including the amygdala and the hippocampus, where emotion and memory can impact our motivation for behavior. And these behavioral systems then are integrated and can produce motor output, not from the posterior frontal lobe, where voluntary goal-oriented behaviors are planned and initiated, but rather through deeper parts of the ventral forebrain where motivations come from. Okay, lastly, as we continue to provide a broad overview of our motor systems, I want to talk about these structures that we find over here to the right-hand side of this figure, the basal ganglia and the cerebellum. Now, the basal ganglia and the cerebellum are not considered to be upper motor neuronal systems. You might wonder why. Neither the basal ganglia nor the cerebellum connect to muscle, so that might qualify them to be called upper motor neurons. But the basal ganglia and the cerebellum, they don't project directly to those circuits that govern the output of our lower motor neurons, which is the usual way that we define what constitutes an upper motor neuronal system. Rather, the basal ganglia and the cerebellum, they interact directly with networks of upper motor neurons. So notice that the arrows enter this block diagram at the level of our upper motor neuron systems. Okay, so we'll see how this works in subsequent tutorials. But for now, let me simply say that the basal ganglia is essentially involved in the initiation and suppression of movement, whereas the cerebellum is involved in the coordination of ongoing movement. We'll see an important role for the basal ganglia just as movements are getting started. And now, once the movements are ongoing, then the cerebellum has a critical role to play in coordinating their ongoing activity. Okay, well, let's now turn our attention to the organization of our lower motor neurons, and we will eventually be talking about our segmental reflexes that are coordinated at this level of lower motor neurons in the spinal cord and brainstem. This figure shows us a Nissl stain section through the lumbar level of the human spinal cord. And if you've not yet taken the time to see my overview of the internal anatomy of the spinal cord, this might be a good time to pause and have a look at that particular tutorial. I think that will help you out with some aspects of where we're going to go over the next few minutes. If you have seen that, great. Let's go ahead and move on. So I won't go through the detail of the organization of the gray matter and the white matter of the spinal cord. Rather, I'll draw your attention to the ventral horn and to a set of columns of neurons that we call laminae 7 through 9. And it's these columns that contain our lower motor neurons. And even at this lower magnification, I think you can see that in this region of the ventral horn, we have these large neurons that are present. And of course, I'd like to show you those in more detail. And so just to give you a higher magnification view of what they actually look like, here we see some lower motor neurons, and they have a very characteristic morphology to them. They, first of all, are very large cells. They have sort of a multipolar shape. This is a Nissl stain, so we're not seeing all of the dendrites that are extending off in various directions. But what we see is a cytoplasm that's just full of darkly staining Nissl substance, which is indicative of rough endoplasmic reticulum and the production of proteins. We see quite a large nucleus and a very prominent and single nucleolus near the center of that nucleus. So this is the characteristic appearance of a typical motor neuron. Here we have another one. The plane of focus is just a little bit off the nucleolus, but I think you can see the nuclear membrane there and then the nucleolus here in the center. Well, that's what the motor neurons actually look like. How are they distributed in the ventral horn is the topic that I'd like to spend just a few minutes discussing with you next. Now, you already know that along the long axis of the spinal cord, there are two regions in which the diameter of the spinal cord is significantly enlarged. There's the cervical enlargement and the lumbosacral enlargement. And these enlargements reflect the additional circuitry and the additional numbers of neurons that are in those segments of the spinal cord that serve the upper extremities and the lower extremities. Well, that's what's going on along the length of the spinal cord as we look at the gross organization of this structure. If we look at a finer scale, what we see are that within the cervical enlargement or within the lumbosacral enlargement, the neurons that supply a given muscle are also extended over some distance across multiple segments of the spinal cord. And we see in this figure the results of an experiment that demonstrates this point. So what was done in this experiment is that the gastrocnemius muscle in an animal model was injected with a dye substance that is taken up at the neuromuscular junction by the axons of the alpha motor neurons that innervate that muscle. And then this dye is transported back to their cell bodies, and then the experimenters can look into the spinal cord in histological sections and plot the location of the cells that were labeled by injecting a specific muscle. Well, the same kind of experiment was also done for the soleus muscle on the opposite side of the body. And as a result, what we see is a column of cells on one side of the spinal cord that reflects the distribution of motor neurons that supply the gastrocnemius. And on the other side of the spinal cord are the labeled motor neurons that innervate the soleus muscle. And the point here is that for each muscle, there is a column of cells, perhaps not unlike this writing device that I'm holding, that is distributed across multiple segments of the spinal cord. And when we look at their positions, we find that they're not overlapping necessarily. There may be some order to their distribution in the medial to lateral aspect of the ventral horn. In fact, this is a very important principle of ventral horn organization that I want to stress for you. If we look at the arrangement of these columns of motor neurons that innervate specific muscles, what we see is a beautiful and hopefully memorable somatotopic organization. Again, I'll use this word somatotopy that we talked about when we considered the somatic sensory system to refer to the mapping of the body. So we see a kind of somatotopy now in the organization of our lower motor neurons in the ventral horn of the spinal cord. What we see are neurons that are present in the medial part of the ventral horn are supplying muscles that are found in the more proximal portions of the extremities. or perhaps in the trunk muscles themselves if we're talking about the thoracic spinal cord. Whereas as we get to progressively more lateral positions in the ventral horn, we move from proximal to distal, such that the muscles that govern the movements of the distal extremities, which is where so much of the skill is expressed, especially via the activities of our arms and hands, these motor neurons are found out here in the lateral part of the ventral horn. So it's very important to recognize this medial to lateral somatotopic organization of the ventral horn, because that will provide an important framework for understanding the descending projections from upper motor neurons. We'll recognize that the descending projections that reside in this ventral or anterior white matter of the spinal cord are mainly supplying the medial aspects of the ventral horn, whereas the major fiber bundle that sits out here in the lateral column of the spinal cord is the lateral cortical spinal tract. And this system is mainly concerned with supplying the lateral aspects of the ventral horn. So getting back to our general organization framework then, I hope you can recognize that what's going on in this ventral and anterior white matter is mostly concerned with setting the stage for skilled behavior in adjusting posture, setting gain, and otherwise facilitating what we do with our distal extremities. And what's happening out here in the lateral part of the spinal cord is mainly an expression of skilled behavior. Now, I won't spend much time right now reviewing for you again the organization of the motor elements of our cranial nerve nuclei. Rather, I'll just refer you back to the tutorial regarding the internal organization of the brainstem and the organization of the cranial nerves and their nuclei. And there what we talked about was the outflow from somatic motor and brachial motor nuclei that are concerned with moving muscles derived from somites or from the pharyngeal arches, respectively. And these motor nuclei are found in each of the three major divisions of our brainstem. In the midbrain, we have the oculomotor nucleus and the trochlear nucleus, and then in the pons, we have the abducens nucleus. These three somatic motor nuclei govern the movements of the eyes in the orbit. And then down in the upper part of the medulla, we have the hypoglossal nucleus that is concerned with the movements of the tongue. And with respect to governing those muscles derived from the pharyngeal arches in the pons, we have the trigeminal motor nucleus that innervates the muscles of chewing. We have the facial motor nucleus that supplies the muscles of the face that are involved in facial expressions of emotion. And then further down in the brainstem, in the medulla, and in the upper cervical segments of the spinal cord, we find first the nucleus ambiguus, which is governing the muscles of the pharynx and the larynx, and in the upper cervical cord, the spinal accessory nucleus, which is innervating the pharyngeal arch-derived muscles that turn the head and shrug the shoulders. Well, I do want to at least show you what these motor neurons look like, just to illustrate the point that in the cranial nerve motor nuclei, we have alpha motor neurons, very much like the motor neurons that we find in the ventral horn of the spinal cord. This is a detailed look at one of our brachiomeric motor nuclei, the trigeminal motor nucleus, and here is roughly the boundaries of this nucleus. And so this is where we would find our columns of cells that are providing innervation to the muscles of mastication, those muscles that we use when we chew and consume food. And what we see are beautiful demonstrations of alpha motor neurons that have all the same histological features as what we saw in the ventral horn of the spinal cord. And lastly, what I'd like to do in this tutorial is to talk about the motor unit and describe for you just some of the physiological features of musculoskeletal behavior that we want to keep in mind as we move into a deeper discussion of how the nervous system controls movement. So, the alpha motor neuron of the spinal cord is what we've been talking about, and here's an illustration of an alpha motor neuron sitting in the ventral horn of the spinal cord. And it sends its axons out through the ventral root into a spinal nerve, and then as that spinal nerve supplies a muscle, this axon will divide into several branches, and these branches will then go on to supply innervation to a set of muscle fibers within a muscle. And these muscle fibers tend to be broadly distributed within that muscle. So the motor unit is defined as the single alpha motor neuron plus the muscle fibers that it innervates. That's the motor unit. Now, a single motor unit can be classified not just anatomically, but also physiologically. There's coordination between the neuron and the muscle fibers for the functions that that motor unit must perform. So from a physiological perspective, there are biochemical and even neural specializations that allow these three types of motor units to contribute to physiological activities of entire muscles in distinct ways. First, we have a type of motor unit that we classify as being slow. This is a motor unit that doesn't generate a whole lot of force, and it does so fairly slowly. So what we're looking at is the response to a single action potential that is applied to the axon that innervates a particular set of muscle fibers. Now, if that action potential arrives at a different collection of muscle fibers, we might see a more significant amount of force generated, and it might happen more quickly. This we call the fast fatigue resistant motor unit. And then if we apply that action potential to yet a third motor axon, we might find a much larger amount of force generated very rapidly by the contraction of a muscle fiber that happens to be innervated by that axon. If we saw that kind of physiological profile, we would know that we're dealing with a fast, fatigable motor unit. Now, if we applied a series of pulses, not just an individual shock, but a train of pulses, we might find something that looks quite similar, except now we would see evidence of sustained response. And so with the activation of the slow motor unit, notice how each individual contraction takes some time, and we build up our steady state level of force production and can sustain that over the duration of the brief train of pulses that is applied in this experiment. The fast fatigue resistant motor unit does considerably better with force production, as can be seen, and notice that it takes much less time to generate that force. And I want you to notice what happens when we get to our fast fatigable motor unit. Of course, this is going to give us the greatest amount of force generated with activation of the axon that supplies this set of muscle fibers. And the force that's generated happens very rapidly. But notice that even over the duration of about a second or so of stimulation, there is some decrement in the amount of force that can be sustained over time. And so it's for this reason that we talk about the fatigue profile of these motor units. And that, of course, can be seen if we extend our stimulation not just over a second or so, but over many minutes. And what we find is that our slow motor units are those that can maintain their maximum amount of force production for a very long period of time. Not indefinitely, of course, but for a long period of time. So, again, the amount of force that's generated is small, and the speed at which that force is generated at a pulse-by-pulse basis is relatively slow. Our fast fatigue resistant motor units do considerably better. They do fatigue, but there is a healthy amount of sustain to the amount of force generated over a long period of time, at least over several minutes. Now, if we look an hour later, now we see evidence of the fatigue that is set in and the ability of these motor units to keep pace with the commands that are coming down the axon. Lastly, our fast fatigable motor units are those that generate the most force, and they do so very quickly, but they're really only good for a few minutes of activity. They fatigue quite rapidly, and after just a couple of minutes of activity, they're just not able typically to sustain the commands that are arriving down their axons. Now let's put all this together in terms of behavior. These are data from an animal experiment looking at the transition from standing all the way up through a more explosive type of motor activity, jumping. And what we see is that to maintain posture, we rely on our slow motor units, and that makes good sense because speed is not an issue. Typically, the amount of force that's needed to maintain posture is relatively low, and we certainly want to activate motor units that are resistant to fatigue. And so as we transition now from maintaining a steady posture while standing to actual locomotion, now we engage next our fast fatigue resistant motor units. This will allow us to generate more force, to do so at a moderate pace that's necessary for a transition from standing to walking, and even progressing through an increase in pace of locomotion. But once we get into a full-out run, or in this animal model, a gallop, and certainly as we approach the most explosive of our motor outputs, we have to engage our fast fatigable motor units, but we do so only strategically. So this sequential progression from slow to fast fatigue resistant to fast fatigable motor units is called the size principle, because it's the principle through which we recruit motor units of different sizes. Now, a point I probably should have made in the previous slide, one reason why our slow motor units generate a small amount of force is that they tend to be the smallest of the motor units, associated with just a few number of muscle fibers. Now, these muscle fibers do have biochemical specializations that also limit the amount of force that each fiber can generate, but the size of the motor unit tends to be small. Whereas the fast fatigable motor units, these are the largest motor units that we have. And recall again that when we talk about the size of the motor unit, we're talking about the numbers of muscle fibers that are innervated by a single alpha motor neuron. And in the case of our fast fatigable motor units, there can be several-fold more muscle fibers innervated than what we find in a typical slow motor unit. And those muscle fibers that are supplied by these fast fatigable motor units are biochemically specialized to generate a maximal amount of force very, very quickly. So this size principle provides an important advantage for coordinating the output of our motor units. It allows for a matching of the physiological properties of different types of motor units with the demands of the specific behavioral task. Now, that's not to say that this plan for coordinating the outflow from our lower motor neurons to these specific types of motor units is not subject to change. It is. Even these muscle fiber characteristics that allow a muscle to either resist fatigue or to be susceptible to fatigue, even those biochemical properties are subject to plasticity, as are the physiological properties of the motor neurons that they innervate. Now, this is a matter for exercise physiology, and we won't go into much depth at all, but let me suffice to say that it is possible to slow the physiological properties of our motor unit. This is one of the principal impacts of endurance training or aerobic exercise on the physiology of our motor units. Well, let me pause there and we'll take a break, and we'll come back with our second part, where we will consider the organization of the circuits of the spinal cord as they express reflexive activity. Welcome back to this tutorial on lower motor neuronal control of movement. In this part, I'd like to talk about segmental reflexes of the spinal cord. So the learning objectives that pertain to this part of our tutorial are several. I want you to be able to discuss the critical components of the myotatic reflex and how they interact to monitor and adjust muscle length. I want you to be able to characterize the role of gamma motor neurons in the adjustment of the gain of our muscle spindle system. I want you to be able to discuss the factors that account for muscle tone. I want you to be able to discuss the critical components of the Golgi tendon reflex and how they interact to monitor and adjust the force of muscle contraction. We'll spend a bit of time talking about the difference between the contributions of the muscle spindle and the Golgi tendon organs to the control of muscle activity. So that will be a key point of differentiating learning objective number 2 from learning objective number 4. And then finally, I want you to be able to discuss the critical components of another type of reflex called the flexion or the cross extension reflex, and how these components interact to withdraw a limb from a harmful stimulus. Okay, so let's begin by talking about a reflex, the simplest of all reflexes that we have through the circuits of our central nervous system. It's called the myotatic reflex. Perhaps you better know this as the stretch reflex. The stretch reflex, or the myotatic reflex, is a system that monitors and maintains the length of a muscle. And the way this system works is through a beautiful organ that we find within our muscles of our musculoskeletal system, and this structure is called the muscle spindle. So the muscle spindle is a collection of muscle fibers that are found within a capsule. And so the boundary of these specialized muscle fibers by this capsule allows the anatomist to call these intrafusal muscle fibers. This idea of a fusiform shape describes the shape of this muscle spindle, or rather the capsule that surrounds these muscle fibers. So there are indeed muscle fibers within the capsule, but these are very specialized muscle fibers that are innervated by sensory axons. So here to the right on the slide, we see our muscle spindle, which is this structure bounded by the capsule. And within that muscle spindle, we see some number of specialized muscle fibers, and those would be our intrafusal muscle fibers. And notice that there are a number of axons that are associated with this muscle spindle. There are two types of sensory axons. There's the group 1A afferent axon and the group 2 afferent axon. The group 1A tends to supply the central portion of these intrafusal muscle fibers, which is where the nuclei of these muscle fibers tend to aggregate. The group 2 afferent axon supplies the contractile portions of these intrafusal muscle fibers, which is found away from that more bulbous shaped central region. And rather than wrapping around these muscle fibers in a spiral type shape, which is what happens with the group 1 axons, the group 2 axons, they make a more limited type of ending that we call the flower spray ending. And the flower spray ending is intended to imply to the microanatomist that the terminals look like petals of a flower that are blossoming on the contractile elements of these intrafusal muscle fibers. There's also a motor supply to the contractile portion of the intrafusal muscle fibers, and this is from a small motor neuron that we find in the ventral horn of the spinal cord. This is called a gamma motor neuron. And we'll come back and talk about the critical role of the gamma motor neurons in adjusting the sensitivity of this muscle spindle to changes in the length of the muscle. Let me back up just a moment and say a little bit more about these intrafusal muscle fibers. They basically come in two varieties. So there are a group of morphologically defined muscle fibers within the spindle that we call nuclear bag fibers. And these fibers tend to have a more bulbous collection of nuclei in their central region within that intrafusal muscle fiber. A muscle spindle might have a handful of these nuclear bag fibers, and there may be an equal number, or more typically a smaller number, of nuclear chain fibers. And these are a different type of intrafusal muscle fiber with respect to their morphology, but also their physiology. The morphological difference is that the nuclear chain fibers has their nuclei arranged typically in a single row along the central portion of that muscle fiber. So rather than there being more of a bulbous region that collects as a bag would, a collection of nuclei, rather there's a chain, a single row of nuclei that give this type of intrafusal fiber its name. So in addition to being morphologically distinct, there are differences between our nuclear bag fibers and our nuclear chain fibers with respect to their sensitivity to stretch and how they contribute to our overall capacity of this spindle to monitor the length of the muscle. Imagine, for example, that we are applying a stretch to this muscle. So we're going from, let's say, length 1 to length 2, and we do so over a relatively short period of time. Well, if we were to look at the nuclear bag fibers, what we would find is that the axons that supply those intrafusal muscle fibers will fire a bunch of action potentials, especially during the change in length. And they won't be quite so modulated as we maintain a more stretched length. In contrast, the nuclear chain fibers, and more particularly the group 2 axons that supply them, they would be more concerned with the maintenance of the steady state that's then achieved once that muscle is stretched to length number 2. So again, the nuclear bag fibers, they're concerned with the dynamic aspects of movement, and the nuclear chain fibers are more concerned with reporting the steady state level that's achieved at the conclusion of the stretch. Now, as you've already seen, these muscle spindles are very important in giving rise to signals that we talk about in terms of proprioception, that is, building up an awareness of the movements of the body. And this awareness reaches consciousness via collaterals that enter the dorsal column medial lemniscal system. But there is also an important signal derived from these muscle spindles that is supplied to the cerebellum. So recall that the cerebellum receives input via a spinal cerebellar pathway about the movements that the body is actually making. Well, this is one important source of information that is supplied to the cerebellum. Well, let's consider then how information derived from these group 1A and group 2 afferents is actually handled in the spinal cord. Well, the most common example that I trust you all have experienced at one time in your lives or another is something called the myotactic reflex, or the stretch reflex. And one common way that this is elicited clinically is with the reflex hammer. So what we see here is our typical knee-jerk reflex, where a force is applied to the patellar tendon. And what's actually being done here with the tap of the tendon is a stretch is being applied to this quadricep muscle. As that stretch is applied, it activates our group 1A axons that supply the nuclear bag fibers in the muscle spindle. So I say group 1A because it's a very quick, dynamic sort of stretch that's applied, and that's exactly the kind of stretch that the nuclear bag fiber in the group 1A afferent is going to signal. So this sensory information runs back into the spinal cord. Yes, there are collaterals that enter the dorsal column region that allow us to make use of this information in central processing at higher levels above the spinal cord. But for now, I want to focus in on what is going on in the spinal cord itself that explains the knee-jerk reflex. Well, the output that produces the jerk involves a contraction of this quadricep muscle, right? So in order to have contraction of this extensor of the leg, we have to have a motor output from the relevant motor unit that supplies the very muscle that was stretched. And this happens because the afferent fiber makes a monosynaptic excitatory connection on this alpha motor unit. And so let's just break down those terms. Monosynaptic means one synaptic connection, and it's excitatory. When the action potential arrives at that presynaptic terminal, there's the release of an excitatory neurotransmitter, probably glutamate or something like that, and a very strong synaptic connection exists here, such that the activation of this sensory axon is very likely to lead to a suprathreshold depolarization of that motor unit and the firing of an action potential that leads then to the reflexive contraction of the muscle and the jerk of the leg. Well, in order to facilitate this behavior, things need to be just a bit more complicated. Now, there is also a flexor muscle there in the hamstring region, and in order to move this limb, ideally one would want to diminish the muscle tone that would be present in that hamstring muscle, such that the quadricep can actually extend the leg. Here's where an additional neuron is interposed between the sensory element and the relevant motor element. And that additional neuron is an inhibitory interneuron. So this inhibitory interneuron receives the excitatory input from this 1A afferent and has the physiological action of inhibiting the motor output to the flexor muscle. So this neuron right here then becomes diminished in its output to that flexor muscle. So by decreasing the numbers of action potentials that is supplying that flexor muscle, the muscle tone of that flexor is now going to relax. And this removes the antagonism about this extensor movement of the leg. So even one of our very most simple reflexes that we see in our central nervous system involves the coordination of excitation and inhibition to achieve functional synergy with the activation of one set of muscles and the suppression of an antagonistic set. The result of this coordination is the smooth expression of movement. So let's look at this from a more physiological perspective with microelectrode recordings. What we see is that when there is a tap of the hammer on the patella tendon, we see in our sensory neuron an elicitation of a barrage of action potentials. So that set of action potentials then leads to the activation of our motor neuron that is innervating our extensor muscle. So this is monosynaptic excitation of that motor unit. Now there's also monosynaptic excitation of this inhibitory interneuron. So with the activation of that interneuron, then we see evidence of the suppression of the output to the antagonistic muscle, the flexor muscle. Notice the suppression of activity that we see during the time that that sensory input is sending its signals into the circuitry of the spinal cord. So we call this disynaptic inhibition of the motor neuron that supplies the flexor muscle. Sometimes we call this pattern reciprocal inhibition because there is monosynaptic excitation to the synergists and disynaptic inhibition to the antagonists. So we've been talking about the stretch reflex as we've commonly experienced it ourselves in a clinical examination with a hammer tap to the patella tendon, but I just want to make the point that this reflex is operating all the time. Consider the challenge of holding steady receptacle for some liquid that's being poured into it. So what's going to happen now is, as we want to maintain a steady angle here across our elbow joint, we are going to be adding a load to the distal part of this biomechanical unit. So that increase in load is going to stretch out this biceps muscle, and that stretch is going to be sensed by our 1A and group 2 afferents, and it's going to activate the very same kind of circuits that we just described in detail for the knee jerk reflex, only now these circuits are in the cervical level of the spinal cord, and they will provide output to the biceps muscle, which will cause a commensurate contraction across that elbow joint that will allow us to maintain this angle at a steady state position, even while the load is changing. And all of this is going to require the activation of synergistic muscles, and it's going to require the disynaptic inhibition of output to our antagonistic muscles, in this case our triceps. So let's see how this works. So now we are adding a passive stretch, which is due to the increased load that's being placed here on the distal part of the extremity. That's going to provide for a stretch signal in the muscle spindle and the communication back to this circuitry via the 1A and group 2 afferents, and that leads to monosynaptic excitation of the motor output to the synergistic muscles, and it's going to lead to disynaptic inhibition of the motor outflow to the antagonistic muscles. And this allows for the increase in resistance and the restoration of the joint angle that keeps us from spilling our beverage. So this is an example in everyday life of how this circuitry works. So in a more schematic form, we have some kind of goal that is set by our motor cortex, and that goal is communicated down through circuits that we'll describe over the next several tutorials. And so this allows the output from an alpha motor neuron to a skeletal muscle to help achieve the goal of steadying this receptacle against a changing load. So as some disturbance is now added in the form of liquid coming into this receptacle, there's a change in muscle length, which is sensed by our muscle spindles, and now afferent activity is fed back to the relevant alpha motor neurons that then increase resistance against this changing load. Now, not depicted in this schematic is the disynaptic inhibition that allows for relaxation of the antagonistic muscles, but that too is part of the distribution of this incoming sensory information to this spinal cord circuitry. So far we've been talking about the operation of this stretch reflex under pretty much one set of circumstances, but there may be a variety of circumstances that would demand a change in the sensitivity of these receptors. So it makes sense that there ought to be some way to change the sensitivity of these receptors even during an active movement. And this is where the input from the gamma motor neuron to the contractile elements of the intrafusal muscle fibers comes in. So let's consider how this works. First of all, what do I mean by gain? Well, what do I mean by gain is simply this. Gain is the amount of extrafusal muscle contraction that can be elicited by a given stretch or a given load that's applied to that muscle. So it's basically an assessment of the output of the motor unit relative to the input in the form of a sensory signal that is monitoring the length of the muscle. So if gain is low, then one can imagine that a moderate stretch will induce only a rather modest output from the central nervous system, leading to only a small increase in resistance given a particular load that's just been applied. If gain is high, then you can imagine that the very same sensory stimulus will now elicit a much greater number of action potentials in the motor output to the muscle that was stretched. and a much larger increase in resistance given the very same sensory stimulus. So gain is a way of either dialing up or dialing down the volume, if you will, of this stretch reflex circuitry. So how is gain adjusted? Gain is adjusted by the output of our gamma motor neurons to the contractile elements of our intrafusal muscle fibers. So one way that we can think of how this works is that activating our gamma motor neurons resets the sensitivity of the muscle spindle. And this would be especially important if the muscle is actively contracting, meaning that the muscle spindle is getting shorter. Well, if it gets shorter, then it loses its sensitivity to monitor the change in muscle length. So let's look at this figure and see how this might actually work. Now, for the purposes of this schematic, we're taking the intrafusal muscle fibers out of the muscle. Of course, they're actually embedded within the muscle, but for this demonstration, let's just simplify by removing these intrafusal muscle fibers. And what we can see now is that if we have a microelectrode that can stimulate the extrafusal muscle fibers, then this muscle can contract against some load. And if we do nothing to the muscle spindle, what we would see is that during the active contraction, the muscle spindle would grow silent. And that's because there is shortening of the muscle. And as the muscle shortens, then this intrafusal muscle fiber becomes somewhat lax. And that reduces the stretch that would be applied and reduces the sensory outflow from that group 1A or group 2 afferent that happens to be supplying that muscle spindle. Now let's imagine that we repeat this experiment, only now, while we stimulate the alpha motor neuron, we also stimulate the gamma motor neuron. So here's the stimulation of the alpha motor neuron and concurrent stimulation of the gamma. So notice what's happening here. As this muscle is contracting and getting shorter, the activity recorded from this spindle afferent is filled in. And so this muscle spindle can remain sensitive to the dynamic aspects of length change, even while that overall muscle length is getting shorter. Now, these adjustments of gain, they can happen in lots of different contexts. In fact, they are probably happening all the time, and this can be highly adaptive because we move in a variety of situations with a variety of goals in mind. So the adjustments of gain are very much context dependent. So the gamma motor neuron activity can be adjusted relatively independently from the excitability of the alpha motor neurons. And this can largely be accomplished by descending inputs that are coming down mainly from the brainstem. We'll talk more about that in a subsequent tutorial. But note that the gamma motor neurons are key to adjusting the sensitivity of the muscle spindle. And that adjustment of sensitivity is itself something that can be governed from above. And you might imagine that this could be very important as we engage, for example, in sport, or perhaps as we're engaged in circumstances that require some sense of fight or flight. We can imagine that an increase in muscle spindle sensitivity is very good for generating explosive kinds of motor activities. So, for example, imagine being a sprinter on the blocks, ready to go when the starter pistol ignites. So we are ready, we're set. Well, we imagine that the gain of our muscle spindle system is probably increased during those moments of getting ready and set, because what we want to be able to do is to monitor a change in muscle length that might happen during this explosive outburst of motor activity that a sprinter would engage in as they exit the block at the sound of the starter's pistol. Well, that's just one example of how descending inputs can modulate the gain of gamma motor neurons. These adjustments very well may rely on descending inputs from the brainstem from parts of what we call the reticular formation of the brainstem that generate and then release in the spinal cord biogenic amine neurotransmitters. So these are transmitters that we consider to be neuromodulators because they have the effect of either increasing or decreasing the excitability of their postsynaptic targets. This is exactly what we might need if we're going to adjust the excitability of our gamma motor neurons, depending upon context. There's even evidence to suggest that the presynaptic terminals of our group 1A afferents themselves are subject to synaptic inhibition, and this would be a means of cutting off the incoming sensory signal independently of whatever adjustment of gain we might employ at the level of the gamma motor neuron. So either by adjusting the sensitivity of the spindle or perhaps interrupting the incoming sensory signals derived from that spindle, we can make the overall sensitivity of this muscle spindle system and its impact on the extrafusal muscle fiber something that can be exquisitely controlled. I think the power of gain control can be easily demonstrated. If any of you are athletes or you participate in stretching activities either before or after some sport or some kind of activity involving exercise, you may be wondering, well, how is it that we can voluntarily stretch? I mean, maybe some of you do yoga or something like that. The fact that you can voluntarily stretch a muscle implies that there must be some kind of way of adjusting the gain of those muscle spindles, because obviously you actually want to lengthen those muscles. You don't want that spindle to contract against your voluntary desire to lengthen that muscle. So that implies that there must be some descending control that will adjust either the gain of the muscle spindle via changing the excitability of the gamma motor neuron and or interrupting the incoming afferent signal that's arriving via that group 1A or group 2 afferent in the spinal cord. All of these considerations that we've been talking about so far pertaining to the muscle spindle are really critical for maintaining muscle tone. So muscle tone is simply the resting level of tension in a skeletal muscle. And this tension depends upon the firing rate of the alpha motor neurons that supply the extrafusal fibers of that muscle. The activity of those alpha motor neurons is what's regulated by the sensory feedback that's coming from the muscle spindle. And as we've already discussed, the gamma motor neuron then will be really critical for adjusting the sensitivity of that muscle spindle and therefore the sensory signal that is then impacting the output of the alpha motor neurons and therefore the tone of the muscle. You can imagine that if there is damage to the alpha motor neuron itself or the axons in the ventral root or the spinal nerve, there should be a catastrophic collapse of muscle tone, because those muscle fibers no longer are receiving their electrical signal from the central nervous system to maintain their resting level of tension. One might also imagine that there would be a catastrophic loss of muscle tone if there should be a disruption of the incoming sensory signals via the dorsal root. And that incoming sensory information is essential for maintaining the output of those alpha motor neurons. So damage either to the ventral horn, the dorsal horn, the ventral root, or the dorsal root, or perhaps the spinal nerve itself, all could produce a catastrophic loss of muscle tone. We call this hypotonia, or sometimes we call the muscle itself flaccid to refer to this catastrophic loss of muscle tone. Now, we'll see in a future tutorial that if there's damage not to this reflex circuitry of the spinal cord, but to the inputs that are arriving from higher levels, such as the brainstem or the motor cortex, one can eventually see exactly the opposite response, an increase in muscle tone. It's as if the gain of these segmental reflexes is turned up by a loss of descending inputs. And this difference between seeing loss of muscle tone or an increase of muscle tone will be absolutely critical for your assessment of the localization of injury based on an examination of skeletal muscle and the status of our segmental stretch reflexes. Now let's turn our attention to another very important sensory element that we find and associated with our musculoskeletal units, and this is called the Golgi tendon organ. The Golgi tendon organ and the spinal reflex derived from its sensory signals provides us with a system that monitors the force that's generated by a contracting muscle. So the stimulus then that activates the Golgi tendon organ is tension. That is the force that's generated during the active contraction of the extrafusal muscle fibers. The Golgi tendon organ is relatively insensitive to the stretch that is applied to a skeletal muscle, and that's because most of the stretch is absorbed by the muscle fibers themselves. The Golgi tendon organ is a structure that's found not in the muscle fiber per se, but in the tendon that attaches the muscle fiber to the skeleton. So what we find in this region of this musculoskeletal attachment is a set of nerve endings that come from a sensory axon called the group 1B afferent neuron. And these endings are woven into the fabric of collagen fibrils that we find here in this region of the tendinous attachment to a muscle. And this anatomic relation between the nerve elements and the collagen fibrils make this structure activated as tension is created. So as these muscle fibers contract, they generate a force that tugs on this region of the Golgi tendon organ, and that causes a deformation of these terminals that are woven in among these collagen fibrils. And that deformation opens stretch-sensitive ion channels that result in the influx of cations and the generation of potentials that, if greater than threshold, can lead to an action potential that then propagates into the central nervous system. Now let's look into the spinal cord and see the circuitry that we find that receives this incoming sensory information. Now, we can think of this system in operation as a form of negative feedback. The Golgi tendon organ is going to be activated by the tension that's generated as a muscle contracts, and the physiological effect of that sensory signal is going to be to diminish the output of the alpha motor neurons that supply the contracting muscle, and thereby reduce the amount of tension that's generated. So this is kind of a negative feedback system. Now, the physiological effect of activating this Golgi tendon organ is that the output to the muscle that's generating force is going to be diminished. And this implies that there must be some kind of an inhibitory interneuron interposed between the sensory signal and the alpha motor neuron. And indeed there is. This is called the 1B inhibitory interneuron. It is a dedicated type of inhibitory interneuron that is postsynaptic to this 1B afferent. And it makes presynaptic terminals on the alpha motor neuron that's responsible for the force that's being generated by that contracting muscle. So as this 1B afferent is activating this 1B inhibitory interneuron, there is going to be synaptic inhibition and a reduction in the output of that alpha motor neuron. And this is going to diminish the force or the tension being generated by this contracting muscle. Now, as usual, we have a pattern of reciprocal innervation, such that as we reduce the activity of the flexor muscle, we're going to actually increase the activity of the extensor muscle. And this will help stabilize the movement around this particular joint. And so this happens via disynaptic excitation of the alpha motor neuron that innervates the extensor muscle. So in both sets of alpha motor neurons, we have an interneuron interposed between the incoming 1B sensory signal and the alpha motor neurons. However, in one case, we have a sign inverting interneuron, that is, an inhibitory interneuron that takes the excitation coming through the sensory arc and turns it into an inhibitory signal that suppresses the motor output. On the other side, on the extensor side, we have an excitatory interneuron that conserves the sign of the sensory signal, that is, excitations coming in from the Golgi tendon organ, and that serves to increase the output of the extensor motor neuron. Well, what good is this system of Golgi tendon organs? Well, we think it's very useful for monitoring the force that's generated by an actively contracting muscle. The activity of this 1B afferent is modulated up or down throughout the course of an active muscle contraction. And we can imagine that this might be very helpful in combating fatigue that might be setting in. We can imagine that as a muscle is fatiguing, it's generating slightly less force. That's going to be reducing the sensory signal into the spinal cord circuitry, and that would diminish the inhibition then in the 1B inhibitory interneuron. And with less inhibition, this might be a way of providing for a reflexive increase in the activity that would help combat that fatigue that's setting in on the contracting muscle. We think that this might also be important if a muscle fiber should be directed by the central nervous system to generate contractile forces that are potentially damaging. So this is a form of negative feedback that would potentially help protect the muscle and potentially its tendinous attachment as well against excessive forces that we might otherwise attempt to generate. Now, like all of our systems of segmental reflexes, the Golgi tendon organ reflex arc is subject to modulation via descending pathways. These descending pathways certainly facilitate various dynamics of human movement that we may choose to participate in. One example might be resistance weight training that some of you may be engaged in, where you might actually want to generate very large forces of muscle contraction, perhaps forces that put you at some risk. And if your Golgi tendon organs are trying to minimize that risk, you can imagine that these descending inputs might actually facilitate the achievement of that very large force of muscle contraction. Certainly any competitive weightlifter would have to have a very well exercised set of descending projections that can interrupt the activity of this Golgi tendon reflex. Otherwise, it would not be possible to generate the forces necessary to be competitive. So let's look at how muscle spindles and Golgi tendon organs might operate in a coordinated fashion during muscle activity. And let's begin by considering what happens when a muscle is stretched against some kind of load that's being applied. So here the muscle is lengthening, and so there's going to be an increase in activity in our muscle spindle afferents as this muscle is stretched. Okay? Now, while that stretch is happening, there's really not much change in the afferent activity derived from the Golgi tendon organ, because again, the Golgi tendon organ is not so much sensitive to a change in muscle length, it's sensitive to the active contraction of the muscle. And so far, this is a passive stretch of a muscle. So that's going to activate the muscle spindle afferent, and it's not going to activate much at all the Golgi tendon organ afferent. However, if we were to now actively contract that muscle by stimulating the alpha motor neuron, what we find is that the muscle is now contracting against that load, it's getting shorter, and if we didn't fill in that muscle spindle afferent by also activating the gamma motor neuron, notice that this muscle spindle now relaxes, and as the muscle is getting shorter, there is a drop in afferent activity derived from the muscle spindle. So this active contraction is going to unload the muscle spindle. However, this active contraction provides exactly the stimulus that will drive afferent activity in the Golgi tendon organ. So as this muscle is getting shorter, there's now a dramatic increase in the output from the Golgi tendon organ. So we can see how during muscle stretch and muscle contraction, the spindles and the Golgi tendon organs are tuned to complementary sources of information about the movement. The muscle spindle is sensitive to length but not tension. The Golgi tendon organ is sensitive to tension but not length. We need them both, and they both operate to allow that muscle to function within the context of the goal that's been established by our descending upper motor neuronal systems. And lastly, in this part of our tutorial, let's consider yet a third reflex, and this is the most complicated of the ones that we've considered, but it's really not so bad once we think about what has to happen here. So this reflex is the one that would be engaged if we were to step on a tack, or perhaps we were to extend our hand and inadvertently touch a hot surface. The immediate response is to withdraw the limb from the damaging stimulus. So this is called the flexion withdrawal reflex, or sometimes it's called the crossed extensor reflex. And while the circuitry involved here looks rather daunting at first glance, I'm going to suggest that it really makes perfect sense if we consider the behavior and what must be coordinated in the central nervous system in order to produce the desired result, which is the withdrawal of the limb from the potentially harmful stimulus. Now let's consider, as is illustrated here, the example of stepping on a tack. Okay, so the cutaneous receptor in question here is going to be a free nerve ending supplied by, let's say, our A delta fiber. So this is going to be an example of a first pain signal coming from an A delta fiber that's transmitted into the central nervous system. And notice what happens. There is the distribution of this incoming sensory signal to a set of interneurons. Well, why does this need to be so complicated? Well, for very good reason. Think of what would be a desired outcome of this sensory stimulus being transmitted into the central nervous system. What we would want to do is we would want to increase the activity of the flexor muscles on the side that encountered the tack. And in order to facilitate the withdrawal of the limb, we are going to want to decrease the activity of the extensor muscles on that very same limb. Now, if we have any hopes of maintaining a standing posture while we withdraw our foot from the tack, we're going to want to do exactly the opposite on the other side of the body. That is, we're going to want to increase the activity to the extensor muscle and decrease activity to the flexor muscle. Now, if you just understand what's going on with the limbs, then I think you can infer what the circuitry must be like in the spinal cord to achieve these goals, knowing that there is what we call polysynaptic activation in the circuitry of the spinal cord. That simply means we have a number of synapses involving several different kinds of interneurons that are interposed between the incoming sensory signal via the A delta fiber and the outgoing motor signal being derived from the alpha motor neurons. And these interneurons need to be, some of them need to be excitatory, some of them need to be inhibitory. In order to activate the appropriate muscles, there needs to be excitatory interneurons linking the sensory signal to the alpha motor neuron. We need to excite the flexor muscles in the ipsilateral limb, that is the limb that encountered the tack. Meanwhile, we need to excite the extensor muscles that are on the opposite side of the body to maintain posture. So that must mean that some of these interneurons must actually span the midline and allow incoming information from one side of the spinal cord to impact the output on the opposite side of the spinal cord. Now, let's look at the actual circuitry that we find here and see what elements that we can detect. So with respect to the flexor muscle on the ipsilateral side of the body, what we have is basically disynaptic excitation. So we have one interneuron, a second interneuron, and then the activation of the alpha motor neuron that supplies the flexor muscle. So disynaptic excitation to the ipsilateral flexor. With respect to the extensor muscle that we want to suppress, we have disynaptic inhibition. That is, we have two synapses, first an excitatory interneuron, and then the signal is inverted by virtue of the fact that we now have an inhibitory interneuron that is responsible for the second synapse between the sensory signal and the motor output. So with that disynaptic inhibition, the output to the extensor muscle is suppressed. Now on the opposite side of the body, we have polysynaptic excitation, again just meaning multiple synapses, perhaps at least two or three are present here, that convey an excitatory signal eventually to the alpha motor neuron that supplies the extensor muscle, and that allows us to maintain our posture even while we are disturbing posture by withdrawing the limb from the tack. Meanwhile, we have polysynaptic inhibition that is applied to the antagonistic muscle on the opposite side of the body. So we have some number of interneurons, maybe 2 or 3, that ultimately lead to an inhibition of the alpha motor neuron that innervates the flexor. So this is a beautiful example of how circuitry can coordinate the output of our alpha motor neurons. And that circuitry can do the job of either increasing the output of the alpha motor neuron or decreasing it, depending upon if the circuitry interposes an excitatory interneuron or an inhibitory interneuron between the incoming sensory signal, which is always going to be an excitatory signal, and then the output from the alpha motor neuron, which likewise is always going to be an excitatory signal. So the way that we increase or decrease the output of the spinal cord to our muscle is by either exciting the alpha motor neuron or inhibiting it through the activity of an interneuron. In the next part of this tutorial, we're going to consider one particular kind of circuitry that will be similar in nature to what we've just described here. It's one that will coordinate the activity of flexor and extensor muscles on both sides of the spinal cord. And it's the kind of circuitry that's essential for generating the rhythmical behavior that supports locomotion. We call this kind of circuit a central pattern generator. So that's the topic we come to next. In this third part of our tutorial on lower motor neuronal control of movement, I'd like to talk about central pattern generation. Our learning objective is simply to describe central pattern generators and their significance for locomotion and other forms of rhythmic behavior. Well, let's talk about locomotion as one example of a basic pattern that is essential for movement, that requires the coordination of the activity of flexor muscles and extensor muscles in our limbs, and even across the midline of the spinal cord. Now, for a quadrupedal animal like a cat, this involves coordinating flexors and extensors in the hind limbs as well as the forward limbs. In bipedal locomotion, which is what we do, we see something quite similar. There's coordination of flexor extensors in our weight-bearing muscles in our lower extremities, but there's also coordination of swing cycles with respect to what we're doing with our arms in our upper extremities. So coordination is happening across the midline. It's happening within the motor columns that are supplying flexor and extensor muscles right next to each other in the same ventral horn. And there's also coordination that's happening across spinal cord segments. So how is this possible? Well, when we just consider the flexion-extension reflexes that are seen when we encounter a potentially harmful stimulus like stepping on a tack, we saw how local circuits can coordinate the activities of flexors and extensors. And local circuits can even span the midline so that what's going on the opposite ventral horn can be synergistic with the behavioral goal. Well, something like that is what we see in the generation of rhythmical movement, rhythmical pattern. And this has been studied for locomotion in mammals, but it's been really worked out quite well in invertebrate systems and in very primitive kinds of vertebrate animals. One such model system that has been very productive in the study of central pattern generation has been the study of swimming behavior in a very primitive kind of vertebrate called the lamprey. Well, the lamprey is a swimming creature that swims through the coordination of a set of segmental muscles, and we now know that the output to those muscles is derived, of course, from alpha motor neurons that send their axons out through ventral roots. But the output of these alpha motor neurons is coordinated through a set of interneurons, some of which are excitatory, some of which inhibitory, and some of which are responsible for generating the essential rhythms that define the swimming behavior. Now, studies of these fairly simple central pattern generators that we see in animals like the lamprey or in other kinds of simple model systems that generate rhythmical behaviors What we find is that central pattern generators tend to have the same kinds of elements, no matter what the model system is that we happen to be looking at. Of course, there are variations on these themes, but we tend to recognize the same kinds of elements that are common to all circuits that generate patterns. One common element is the presence of pacemaker neurons. Some of these neurons will generate the rhythms that define the behavior, and these rhythms reflect the activities in oscillations of membrane potential, in bursts of action potentials that are generated by this set of pacemaker neurons. Central pattern generators typically involve an interaction between excitatory neurons and inhibitory neurons. Some of these neurons are going to send connections that cross the midline, and this allows for the coordination of circuitry in one ventral horn with that of another. There will also be circuitry that spans the segmental levels, allowing for the interactions from one segment to another. Another common element that we see in central pattern generation is modulation from descending inputs. For vertebrates, this comes in the form of descending inputs from the brainstem, and this allows the expression of pacemaker activity to actually be initiated by these descending inputs. Often these inputs release biogenic amine neurotransmitters, and these biogenic amines can often set the rhythm in motion, or they can modulate the rate of the rhythm. There's also input to these pacemaker circuits that come from sensory signals that are derived from our sensory receptors and conveyed via the dorsal roots into the spinal cord circuitry. But these sensory signals often are not essential, so it's possible to remove these sensory signals and still see the expression of this essential rhythm generated by this circuit. And one final feature that I'll mention of central pattern generators is that they can actually generate different kinds of movement patterns. Consider, for example, the difference between a walk and a jog and a run and a sprint. This is not simply just a matter of the rate of output, but there's actually a different pattern that can be produced. This is especially true in quadrupedal animals, where there's a transition from an alternation of activities in the limbs between forelimbs and hind limb to a coordination that is concurrent with the transition from a trot to a gallop. So something like that must occur in our central pattern generators as well, as we transition from one kind of pace to another. So the very same circuit can generate more than one pattern. Although the activity of these central pattern generators can be modulated via descending inputs, the pattern itself can still be expressed even in the absence of these descending inputs. And this point was made rather dramatically some decades ago in experiments done on cats that were subject to transection of the spinal cord. So in the experiments illustrated here, what was discovered is that with a thoracic transection of the spinal cord, it was still possible to suspend this cat on a treadmill and record the rhythmical activities of extensors and flexor muscles that were consistent with locomotion as the hind limbs transitioned between the stance phase, where pressure is applied to the treadmill, and the swing phase, as the limb is removed from the treadmill and then swung forward for the next cycle of step. This basic rhythm of stance and swing with the activation of flexor muscles and extensor muscles was seen even after the dorsal roots were sectioned, suggesting that this is a pattern that is intrinsic to the circuitry of the ventral horn of the spinal cord, even though typically it's informed by sensory signals coming in through the dorsal roots, and it's modulated and initiated via descending inputs from the brainstem. Now, as you might imagine, there's been considerable interest in whether this kind of treadmill training for human beings who've suffered either spinal cord injury or brain injury can be clinically significant for walking recovery. Well, there is ongoing effort to try to study this question. So the upshot seems to be that, at least for walking behavior in people, that our spinal cord seem to be more dependent upon the descending controls for coordinating and facilitating the activation of our central pattern generators than in some of the other animal models that have been explored. Well, we come now to the last part of this tutorial, where we consider the clinical picture of individuals that have suffered damage to their lower motor neurons. So I'll see you in just a moment, and we'll consider the lower motor neuron syndrome. Of lower motor neuronal control of movement by considering the clinical picture of individuals that have suffered damage to their lower motor neurons, to their spinal nerves, or to the circuits that govern the output of those lower motor neurons. This is called the lower motor neuron syndrome. So my learning objective for you is simply to characterize the signs associated with damage to lower motor neurons. So this lower motor neuron syndrome has several components to it that I want you to be able to recognize. If we section the nerve that supplies the skeletal muscle, or if we injure the ventral horn of the spinal cord where the cell bodies of those neurons reside, we will see a response in the muscle tissue. Over time, that muscle will atrophy. But along the way, we might see abnormal contractions of individual muscle fibers and of individual muscle units. And these abnormal contractions we call fibrillations and fasciculations. So a fibrillation refers to the contraction of an individual muscle fiber. A fasciculation would be the contraction of the muscle fibers of a motor unit. And this is a pathological sign. It indicates the disruption of normal signaling that comes from the spinal cord to those muscle fibers. In addition to these changes that reflect the disrupted physiology to the muscle fiber, there are changes that are more directly indicative of damage to the lower motor neuron itself, or the axon that allows that lower motor neuron to communicate to that muscle fiber. And these kinds of signs and symptoms are reflective of the loss of the control of voluntary movement. So there can be muscle paralysis, implying a nearly complete loss of volitional control. Or if the loss of control is less than complete, there still may be profound weakness, and we call this paresis. So the idea is that our brain is sending signals to the spinal cord commanding a voluntary movement like a contraction, and there's profound weakness there. Not because the signal is not being received by the spinal cord necessarily, but rather the output from the spinal cord, or perhaps from the cranial motor nuclei, to the muscle fiber itself is the problem. Now, going hand in hand with this loss of volitional control, or this profound weakness, would be a loss of our myotatic reflexes. So this is what we call areflexia. So there may be a sensory arc that's intact, but without the output from the alpha motor neuron to the muscle fiber, there can be no reflex. And with the loss of volitional control, with the loss of the reflex, we would expect there to be a profound reduction in muscle tone. So this is a devastating condition that is very characteristically observed clinically. So one can expect to see changes in the muscle fiber. One can expect to see evidence of a dysfunction in the reflex arc that supports muscle tone and the execution of voluntary movement. And you should, as you advance in your clinical studies, come to recognize this lower motor neuron syndrome and differentiate it from other kinds of movement disorders. One important differentiation that we'll be making in a set of tutorials that will be forthcoming will be a distinction between the lower motor neuron syndrome and the upper motor neuron syndrome. What you'll discover is that in the hours to days after damage to upper motor neurons, the patient might look very much like a person who has suffered a lower motor neuron injury. But over time, a different set of signs and symptoms will evolve, and that evolution will be a key feature to focus in on. So in addition to thinking about the anatomy that's been injured, you should also recognize the time course of the development of that injury. Even for the lower motor neuron sign, for example, it will take some time for muscle atrophy to set in. You won't see that right away. Likewise, the fibrillations and fasciculations tend to be indicative of the early phases of injury. Over time, those will subside as the muscle fibers become atrophic. So paying attention to the time course of the development of these signs and symptoms and how they may change is going to be a key aspect of your clinical observation. Well, this concludes our discussion of lower motor neurons. The next time we come together, I trust we will be talking about the means by which upper motor neurons govern the activities of our alpha motor neurons and the local circuit neurons that coordinate their output. So next time, we'll be talking about upper motor neuronal control of movement. I'll see you then. Welcome to this tutorial on upper motor neuronal control. This topic, once again, confronts the complexity of the brain, and we come now to consider how movement is controlled by the brain. Obviously, a very complex topic, but one that we are beginning to understand with greater clarity. We're going to be describing circuitry in the brain that is determined by genetic plans and laid down early in embryological development, but nevertheless, life experiences can have a significant impact on this circuitry. And this is a wonderful thing because it allows us to have a flexible motor system that can learn new skills and take on new challenges. And this, of course, is germane to human performance as well as to rehabilitation following impairment of motor control. So this tutorial is going to be broken into several parts. We begin with a general overview of the motor system and the means by which upper motor neurons govern lower motor neurons. And our focus for the remaining portion of this part will be on the primary motor cortex. So the learning objective for this first part is that I want you to be able to discuss the organization of the primary motor cortex and its contributions to the control of volitional movement. I want you to be able to characterize the representation of the body in the primary motor cortex and to compare that representation that we find in the precentral gyrus with what we've already discussed in the postcentral gyrus, which is where we find our primary somatic sensory cortex. And finally, I want you to be able to think well about how movement is encoded in the primary motor cortex. So I want you to consider the phenomenon of population coding as one means by which movement is governed in the cortex. Okay, so let me once again show you this schematic that lays out before us the organization of the motor system. And we've already talked some about our lower motor circuits, which includes the lower motor neurons themselves that innervate skeletal muscle, but also local circuit neurons that organize the output of these lower motor neurons. And those local circuits can integrate segmental reflexes. They can generate rhythms and patterns if they're part of networks that we call central pattern generators. And I've already made the point to you that it's really these local circuits that are receiving this descending input from higher centers in the brain. Although there is some access to lower motor neurons themselves, and that access is seemingly privileged for the parts of the spinal cord that govern the expression of skill, especially what we do with our hands and our fingers. But for the most part, what we're going to be concerned with today is how upper motor centers interact with these lower circuits that help organize the motor output of our motor neurons in the spinal cord and in the brainstem for our cranial nerve motor nuclei. And what we're going to talk about is really two main stations. There are centers in the brainstem that provide for upper motor neuronal control and centers in the motor cortex. So in this part of the tutorial, our focus will be here in the motor cortex, but we'll also spend some time talking about the brainstem in subsequent parts. Now, just to help us understand really the relationship between upper and lower motor neurons, I'll remind you of a phrase, memorable phrase that Charles Sherrington, great British physiologist in the early part of the 20th century, provided for us. He thought of these lower motor circuits as the final common pathway for movement. So to me, what this phrase final common path implies is that it's up to the lower motor circuits to actually implement the movement. And one theme that I'd like to share with you is that what's really going on, especially at the level of motor cortex, is not micromanagement of movement, but rather the encoding of movement intention. And so this will be a theme that I hope to elaborate as we progress through this tutorial. So our focus again here is going to be on the motor cortex, and what we will discover there is really a mosaic of motor control areas. So this mosaic of motor control areas is found in the posterior part of the frontal lobe. So again, to reorient you to the human brain, here's the central sulcus. So this then is the precentral gyrus, and the posterior part of the frontal lobe then would be from the central sulcus, maybe anterior, including the precentral gyrus, and then the posterior part of our superior, our middle, and our inferior frontal gyri. So that's what we're talking about. That's what we mean by this motor cortex, and that's where we would find it, in this posterior part of the frontal lobe. So these motor cortical areas are involved in the expression of what I like to think of as specific motor behaviors or skilled motor behaviors. So these are behaviors that we choose to perform, so there's an intentionality about it, or an aspect of will, our volition. Also included in this motor cortex are areas within this broader mosaic that are concerned with behaviors that, well, perhaps are somewhat less volitional. And what I have in mind here are the kinds of behaviors that relate to the expression of emotion. We can think of the extremes of fight or flight, but we can also think of the nuances of organizing our facial gestures and our posture in a way that communicates non-verbally and perhaps somewhat unintentionally the status of our cognition as it pertains to our sense of well-being, our sense of mood, our affect. So we'll see within this motor cortical mosaic divisions that are more concerned with the expression of emotion than the expression of skilled behavior. So in addition to these motor cortical areas, let me just say a word about these brainstem centers that we'll come to in a different part of this tutorial. These brainstem centers are organized aspects of movement that I like to think of as setting the stage for the expression of skill. If you will, if we can work with this theatrical metaphor, the brainstem works behind the scenes. Perhaps they're the stage managers in the theatrical production, so to speak. The brainstem centers are concerned with adjusting posture, with setting the gain of segmental reflexes. These activities are important so that we can express skill. Now, thinking anatomically, the skill that we expressed is often done with our distal extremities, our hands and our feet, whereas the stage setting functions typically concern our posture, which involves our axial muscles and our proximal limb muscles. So one differentiation between the contributions of the motor cortex to movement and the brainstem have to do with the difference between the skilled behaviors that we express with our distal extremities and also our oral facial structures, and those behaviors that set the stage for the expression of skill, involving our trunk and our proximal limbs. And what I'm thinking about primarily is posture. Now, one important way to think about this differentiation between skill and posture is to recall the way that the body is mapped in the ventral horn of the spinal cord. You will recall that there is a medial to lateral mapping of the body, with the more proximal limb muscles mapped in columns of lower motor neurons that are found near the medial part of the ventral horn, and the distal muscles are governed by lower motor neurons that are found at progressively more lateral positions in this ventral horn. Now, as we turn our attention to the way that upper motor neurons control these lower motor neurons, I would highlight that we see the same medial to lateral organization with respect to the white matter that provides a means for upper motor neurons to communicate with lower motor neurons. Out here in the lateral part of the spinal cord is where we find the lateral cortical spinal tract. And this is mainly going to provide axons that are concerned with governing the control of these distal extremity muscles. So this huge input from motor cortex to spinal cord is primarily directed at the lateral part of the ventral horn. Now, we also have a anterior or ventral and medial part of the white matter that is primarily receiving axons from the brainstem. So these axons are terminating in the medial part of the ventral horn, where we have motor neuronal columns that are mainly concerned with posture and proximal limb muscles. So again, this medial to lateral dichotomy is very important. We can associate this lateral cortical spinal tract primarily with skill, and this medial group of axons that includes components from the brainstem and a small component from the cortex as mainly being concerned with posture and other elements that I'll simply call stage setting. And if you can remember my theatrical metaphor, then perhaps you will recall what is really going on here in these medial white matter systems, setting the stage for the performance of skill. Well, let's have a closer look now, at least in schematic form, at the way upper motor neurons relate to lower motor neurons. Now, this is really a schematic view of the same principles that I just attempted to illustrate for you. Notice that in the lower motor neurons, which are at this level, we've now schematically organized them into medial and lateral components. Okay, so we have the medial cell columns that are found in the medial part of the ventral horn, and then the more lateral cell columns that are found further out lateral. And so that should remind you that the medial columns are concerned mainly with axial and proximal limb muscles, while the lateral columns are mainly concerned with the distal limb muscles through which we express skilled movement. Now, if we look at the way that the upper motor neurons in the cortex and the brainstem relate to these columns of lower motor neurons, this figure is intended to emphasize the point that the upper motor neurons in the cerebral cortex are mainly concerned with the lower motor neurons in the lateral part of the ventral horn. And I want you to notice in this figure the anatomical relationship across the midline of the body. So the upper motor neurons on one side, let's call this the left side, are going to govern the movements expressed via the contralateral side of the spinal cord. In this case, it would be the right side. And so that implies that there must be a pathway that crosses the midline. So we are going to spend a fair amount of time talking about this pathway, and this pathway is in fact that lateral cortical spinal tract that I mentioned just a moment ago. Okay, so the lateral components of our upper motor neuronal pathways in the spinal cord relate to the lateral part of the ventral horn, which is concerned with skill. Now, let's talk about the upper motor neurons of the brainstem. So these neurons are sending axons down through the white matter of the spinal cord, but mainly in this anterior and medial part of the white matter of the spinal cord. And these axons govern the output of the circuits that we find in the medial part of the ventral horn that are mainly concerned with axial and proximal limb muscles. And with respect to sidedness, I want you to know that this input is distributed to both sides of the spinal cord. There are some exceptions where the inputs are primarily to one side or the other, but for the most part we can generalize and say that our brainstem pathways give rise to bilateral projections to the medial part of the ventral horn. In contrast to the inputs from the motor cortex, which are primarily directed across the midline to the contralateral side of the spinal cord and specifically to the lateral part of that ventral horn. So what I've tried to outline for you here is this concept of parallel pathways. So we've talked about this quite a bit in our sensory systems, and now we encounter it in the motor systems, where there are descending inputs from different gray matter structures that are running in parallel and targeting different parts of our lower motor circuitry that we find in the spinal cord. Now there's an even larger dimension of parallel pathway for motor control. So here in this slide we have illustration of how our upper motor neurons in the cortex and the brainstem collaborate in order to express those movements that arise from our volition. And so we have, again, these lateral components that are mainly derived from the cortex, and they're concerned with the fine control or the skilled behavior that we express with our distal extremities. And then there are medial components that are arising primarily in the brainstem, and they're concerned with adjusting posture as well as governing the more proximal limb muscles that are necessary in order to express behavior with the distal extremities. Now there are lateral and medial pathways. The lateral pathways are primarily associated with a major projection from the cortex to the spinal cord that we call the lateral cortical spinal tract. And this is a collection of axons that, as it runs through the brainstem, and specifically in the medulla, forms a structure that we call the medullary pyramid. So for this reason, we call this pathway the pyramidal pathway. Sometimes we speak of a pyramidal motor system. What we have in mind is the connection between the motor cortex and the spinal cord that runs through the medullary pyramid. So the pyramidal motor system is a kind of shorthand to refer to this aspect of volitional motor control that links the cortex with the lower motor circuits of the spinal cord. Now there's another term I'd like to introduce, and that's cortical bulbar. This is a bit of an odd term, I know, but what it refers to are projections from the cortex to the brainstem. This term bulbar often is used to refer to the brainstem, perhaps because of the way that the pons gives the brainstem the appearance of a bulb, perhaps. But nevertheless, the point here is that cortical bulbar is to be contrasted with cortical spinal. Cortical spinal obviously refers to projections from the cortex to the spinal cord. Cortical bulbar refers to projections from the cortex to the brainstem. And for some authors, it means specifically the projections to the cranial nerve nuclei that are expressing motor behavior. So those would be our somatic motor nuclei and our branchial motor nuclei. There are massive projections from the cortex to the brainstem that are not so much concerned with providing upper motor neuronal control. We'll talk about those separately when we consider the cerebellum. So in a subsequent part of this tutorial, I'd like to spend just a little bit of time illustrating for you an emotional motor system. So what I have in mind here are projections that are coming from what we'll call the limbic centers in the forebrain, which are primarily in this ventral medial forebrain, and some other structures, including the hypothalamus and the amygdala, that provide descending projections. Now, I'm going to stretch the medial and lateral concept just a bit, but I think it's possible to apply the same concept of lateral and medial descending pathways that we see very clearly in the control of voluntary movement to the organization of emotional expression. So what we see in our lateral pathways are pathways that allow for the expression of specific emotional behaviors. And what I might have in mind here as examples would be motor behaviors that characteristically define a particular emotion. be it a facial expression, for example, or a posture. Meanwhile, there are medial pathways that set the stage. They're concerned with setting the gain, for example, modulating the threshold at which we express that specific emotional behavior, be it our tendency to laugh or to cry or to become angry, and to express that emotion within the muscles of the face. Okay, well, now with that bit of an overview of the relationship between upper motor neurons and lower motor neurons, let's focus in on the motor cortex. Again, we're going to be considering the posterior part of the frontal lobe, which will include the precentral gyrus, and then the posterior part of our 3 long parallel gyri that we identified in our laboratory lessons. Okay, our superior, our middle, and our inferior frontal gyri. So from both an anatomical and a functional perspective, we can broadly conceptualize the motor cortex as being divided into 2 main regions. There's a primary motor cortex, which is found in the anterior bank of the central sulcus, including much of the crest of the precentral gyrus. And then just in front of that primary motor cortex, we find a premotor cortex. And these areas extend into the midline hemispheric surface, so we can recognize the primary motor cortex as occupying a large portion of that paracentral lobule. If you remember that anatomical term, paracentral lobule refers to this gyral formation that forms the medial cap or the medial terminus of the central sulcus. Just in front of the paracentral lobule, then, is where we find the medial extension of our premotor cortex. And we'll spend just a little bit of time talking about an interesting region right about there that's concerned with the expression of emotion in the face. Now, each of these divisions of the motor cortex, the primary motor cortex and the premotor cortex, give rise to these descending projections to lower motor neuronal circuits in the spinal cord as well as the brainstem. Collectively, these divisions of the motor cortex receive input from the thalamus, as do all regions of the cerebral cortex. We can identify 2 nuclei of the thalamus that are primarily responsible for projections to the motor cortex. They are the ventral anterior nucleus and the ventral lateral nucleus. Sometimes we refer to them together as the VA-VL complex of the thalamus. In addition to input from the thalamus, these divisions of the motor cortex receive important projections from other parts of the cerebral cortex. And one important source of projection comes from the somatic sensory cortex, which sits just posterior to the central sulcus. There are projections from the somatic sensory cortex into this motor cortex, both the primary and the premotor areas. In addition, there are projections from the more posterior parts of the parietal lobe that are processing information about visual spatial relations. Recall that WARE pathway that was processed and elaborated in the posterior part of the parietal lobe. This part of the parietal cortex likewise sends inputs that are relevant to the guidance of movement. So the remainder of this part of the tutorial, I'd like to focus on the structure and function of the primary motor cortex. And this is really a fantastic part of the human brain. If we prepare histological slices, as I did some years ago with my colleagues here at Duke, we can really recognize the fantastic appearance of this part of the brain as it's viewed under the microscope with a standard histological stain like a Nissl stain. And a few features really stand out. One feature, which is highlighted in this slide, is the presence of these very large neurons that we find in layer 5 of the primary motor cortex, which corresponds in Brodmann's nomenclature to area 4. So here in layer 5, we find these very large neurons called Betz cells. In fact, they are the largest neurons in the entire cerebral cortex, and they contribute to the cortical spinal tract. So generally speaking, layer 5 in the motor cortex is what gives rise to projections to the spinal cord and to the brainstem. So we call these connections, again, the cortical spinal and the cortical bulbar tracts. They come from layer 5. Now, just a word about these BET cells. Perhaps when they were first described and recognized, it was tempting to think that these cells are the sole origin of the cortical bulbar and cortical spinal pathways. But we know that there just aren't enough BET cells to account for the numbers of axons we see descending to the brainstem and the spinal cord. So really these BET cells only account for a small percentage of the overall projection from cortex down to lower motor circuits. However, they seem to have some privileged access to those lower motor neurons. I mentioned that most of the connections from cortex to lower motor circuits are actually targeting the interneurons, not the alpha motor neurons themselves. Well, it seems that these BET cells are among those cortical cells that have monosynaptic connections to alpha motor neurons. So they do seem to be given privileged access to govern the output of our lower motor neurons. So you may be wondering, why do we call this Brodmann's area 4 primary motor cortex? Well, it's primary in the following sense. When motor cortex was first characterized with microelectrode stimulation, it was discovered that as one approaches the posterior frontal lobe with the microelectrode and then stimulated different sites, it was possible to elicit various kinds of movement. We'll talk more about that in a few minutes. And the thresholds for stimulation that produced movement got lower and lower and lower as the microelectrode was advanced towards the central sulcus. And the cortex in the anterior bank of the central sulcus was found to have the lowest threshold for producing a movement when stimulated with an electrical current. So this region was defined as being primary for that reason. Now, it was clear from the very earliest stages of these kinds of experiments that one could elicit movements from more anterior regions. So this posterior frontal lobe was considered the location of the motor cortex, but there was a clear distinction in threshold. And so for that reason, this primary motor cortex was identified with the precentral gyrus. There's another sense in which this Brodmann's area 4 can be considered to be primary motor cortex, and that is because it's there in the primary motor cortex where we seem to be especially concerned with the movements around our personal space. For example, bringing the hand to the mouth, or bringing an object close to the visual midline so that we can inspect with our special sensory systems what we happen to be holding in our hands. This kind of movement seems to be well represented and encoded in the activities of neurons in the primary motor cortex. So in this sense, the primary motor cortex can be considered primary because it has the lowest threshold for the elicitation of movement, and it seems to be concerned with those movements that are primarily concerning our bodies and the space immediately in front of our bodies. Now let's talk some about the way the body is mapped out in the motor cortex. So we can recognize a kind of body mapping or somatotopy in the motor cortex. And generally speaking, along the length of the central sulcus and the precentral gyrus, there is in fact a kind of somatotopy. And this somatotopy bears some resemblance to the body mapping that we saw in the postcentral gyrus, such that here on the paracentral lobule, we have representation of the lower extremity, and as we progress towards that S-shaped bend near the center of the central sulcus, we move up the lower extremity through the trunk and into the upper extremity. And near the central part of the central sulcus, where that S-shaped bend is found, that's where we see representation of the movements that we perform with our arm and our hand. And then just lateral and inferior to that region, just as the central sulcus tends to straighten out, we find representation of the face in the inferior 1/4 or 1/3 or so of the precentral gyrus. So this organization from lower extremity through trunk to upper extremity and then to face is something of a mirror image of what we found in the postcentral gyrus. However, there's an important sense in which it's not a true mirror reflection of the fine detailed mapping of the body that we find in the postcentral gyrus. What we find is a much less precise mapping of the body. In fact, we call this fractured somatotopy. Let me give you some examples of what I mean by fractured somatotopy. Here again is this basic layout of the somatotopy in the precentral gyrus from lower extremity to upper extremity to face. And if we simply focus in on the region of the upper extremity, I can illustrate for you why the somatotopic map in the precentral gyrus is not a mirror reflection in detail of the map that we find in the postcentral gyrus. So there's a number of senses in which this body map is fractured. One sense is that we can find in overlapping regions of the precentral gyrus representation of extensor movement and flexor movement. One might think that these would be very different kinds of movements that might have differential columnar representation in the motor cortex. Well, that may not be the case. There seems to be an overlap in where we can find sites that, when stimulated, would produce extensor activity or flexor activity. Another sense in which this body map is not a mirror reflection of the smooth and contiguous mapping of body part that we find in the postcentral gyrus is that we can find multiple representations of the same body region. So, for example, one could probe an area of the precentral gyrus and discover that there is a collection of cells that, when stimulated, move the first digit. Well, one might find a different location, probably not far away, but a different location nevertheless, that likewise, when stimulated, can produce movement in that same body part. So we can have multiple representations in addition to overlapping representations of different types of movement. And lastly, if we look within this region that represents the upper extremity, what we can find is an interdigitation of sites that, when activated, can move, for example, the wrist. And that might be right next to a site that, when stimulated, can move a digit. And those regions might be adjacent to shoulder or perhaps elbow flexion or extension sites. So there seems to be no strict internal somatotopy within this general region that represents the arm. And it's for these reasons that I elected not to illustrate the somatotopic representation of movement in the precentral gyrus with a homunculus-style cartoon, because I think that would be misleading. So you might get the impression that there is a detailed somatotopic mapping of the body in the precentral gyrus. And that really would be misleading, I think, because of these various features of motor representation. What we find is more a general representation of body region that roughly corresponds to the detailed map we find in the postcentral gyrus, but it is fundamentally different in its internal organization. And I think this is quite interesting because what this arrangement allows for is the organization of functional ensembles of neurons for the performance of particularly meaningful behavior. The circuitry seems to be dynamic and flexible and plastic, and that circuitry may provide us with a neural substrate for the acquisition and storage of motor skill. So if this is the means by the motor cortex is represented, it makes some sense that we would see some reflection of this in the connections between motor cortex and lower motor neurons, and indeed we do. If one were to trace, for example, the distribution of a single axon in the cortical spinal tract, what you might find is that that axon terminates in multiple columns of lower motor neurons in the ventral horn. That might be consistent with a single location in the motor cortex being concerned with multiple kinds of movements. Also, if one were to look physiologically, one might find that a single action potential or a single spike in a cortical spinal axon might activate different muscles in the forelimb. So these 2 points sort of go together. If a single axon can innervate multiple columns of motor neurons, it makes some sense then that one would associate the activity of that single axon with movement of multiple muscles. So these anatomical features and these physiological features at the unitary level, I think, are a reflection of what we find at the level of MAPS in the precentral gyrus. So if all of this sounds very complex and very confusing, I can assure you it is, and we're still trying to understand exactly what is mapped in the primary motor cortex. So I hope that's a question that is raised in your mind at this point in our discussions. So what is represented in the primary motor cortex? It doesn't seem to be a strict representation of the body and its muscles. So I think what we can conclude then, based on the evidence to date, is that what's represented in the cortex is movement, not muscle. In fact, we might go so far as to hypothesize that truly what's represented is movement intention. And I'll give you some examples of what I mean by that. Now, we do know from a long history of investigation in this part of the brain that there are various dimensions of movement that are represented in the motor cortex. For example, the amount of force that we generate with muscle contraction, the directionality of movement, if we're talking about moving an articulated musculoskeletal structure like the upper extremity, the amplitude of the movement, whether it's a small movement or a large movement. These features, these dimensions of movement, seem to be encoded in our cortical network. But we also seem to have representation of movements that are ethologically meaningful. For example, the primary motor cortex seems to be concerned with coordinating movements of the hand and the lower part of the face. For example, moving the hand to the mouth. That's one aspect of movement that seems to be organized in the primary motor cortex. And perhaps that's one advantage of having the representation of the face adjacent to the representation of the hand in the body map. We also see evidence that organized in the motor cortex is representation of skilled manual behavior in the space that's right in front of us, what we call central personal space. This might be very important for bringing objects into our near visual field so that we can inspect and examine them. Imagine the importance, for example, of having a careful look at a piece of fruit to make a judgment about whether it's truly palatable or not. Well, with all of this being represented in the primary motor cortex, we can make some predictions as to what we might find in a patient that has had damage to this part of the brain or to the pathway that emanates from it. Generally speaking, what we find is that lesions of the primary motor cortex impair the fractionated or the skilled movements of our distal extremities and the lower part of our face. So, for example, some kind of fractionation movement of the fingers of the hand would likely be impaired with damage to the primary motor cortex. So I want to come back just for a minute and say more about my first point here, that what's encoded in the primary motor cortex may very well be the intention of the movement. So I think a really interesting series of studies were performed by Michael Graziano and his colleagues, where they were probing the movements that could be generated in the brain of a rhesus monkey when the motor cortex was stimulated. One of the interesting features of this study that I think make it especially noteworthy is that the duration of stimulation was longer than is typically done in the kinds of body mapping experiments that were done throughout the 20th century. Rather than stimulating for just a very brief moment in time, Dr. Graziano and his colleagues decided to extend the period of stimulation to a period of a few seconds, which was thought to mimic the more natural evolution of activity that emerges in the motor cortex during the performance of motor behavior. And what was found is that microstimulation of certain sites in the motor cortex produced behaviors, not just movements, but behaviors. And here's one example. What we're looking at here is a record of movement with stimulation of the hand region of the precentral gyrus in this rhesus monkey. And the blue plus signs indicate the starting position of the hand at the onset of stimulation, and then the red circles indicate the end position of the hand at the termination of the movement that was elicited with microstimulation. And I think what's obvious here is that at the particular site of stimulation, what was observed is movement of the hand from peripheral regions, be they to the left or to the right of the midline, it didn't seem to matter. And what we found here is hand to mouth movement. So wherever the starting position of the hand happened to be at the time of stimulation, it was brought towards the mouth. And it seems as though in this particular location, what's really important are the kinds of movements that would be needed for feeding. When Graziano and his colleagues moved the microelectrode in a slightly different direction in the primary motor cortex, what was discovered is a site where the hand was moved not to the mouth, but more towards this central personal space where an animal could inspect through vision and presumably other special sensory systems just what was being held in the hand. So these kinds of studies suggest that what's represented in the primary motor cortex is movement intention, bringing the hand to the mouth or bringing the hand to central midline for visual examination. So how might such movement be encoded in the firing patterns of neurons in the primary motor cortex? Well, one idea might be that for each particular movement that is illustrated here with a black line, there may be a neuron that is specifically tuned for the expression of that movement. Well, that would be one idea, and it turns out that that is highly unlikely. Rather, what we find is encoding not by one neuron at a time, but by populations of neurons, and encoding for the various dimensions of movement that I alluded to earlier, the direction of movement, the force that's generated, the amplitude of the movement. And I'd like to show you an additional experiment done some years earlier that really made this point that movement direction is encoded in the population activity of the motor cortex, not by any single neuron. So here's a simple experiment that's being illustrated here. There is a monkey that's trained to move a central joystick in the direction of some kind of target, and that target might be a green light that would illuminate at a particular location in this apparatus. So there's a particular directionality to the movement. And the question then is, well, how does the motor cortex encode this directionality of movement towards a visual target? And the answer seems to be that the encoding is done at the level of the population. So what we're looking at here are a series of raster plots that report the action potentials that were recorded during particular trials. So each row that we see here, we have 1, 2, 3, 4, 5 rows. These are 5 trials. And then each little tick mark that we see represents the occurrence of an action potential. Now, as the monkey moved the joystick towards various visual targets in these various directions, what was generally observed in the firing of individual cells in the motor cortex is that they tended to respond very broadly with movements directed towards the left and in the upward direction. So these movements that are represented by this yellow wedge of this circle here are those that led to an increase in the firing of action potentials around the time of the movement. The movement is represented by this red line here, which indicates at time 0 when the movement actually occurred. So the firing of this cell seems to increase a few hundred milliseconds before the movement was made and continues during the course of the movement. Now notice what happened as the animal moved in the opposite direction. So as the animal moved broadly to the right, what we find is a suppression of activity just before and during the course of the movement. So there seems to be a very broad measure of tuning that can be reported at the individual neuronal level. In fact, if we look at the peak responses as the animal moves in different directions, what we see is a very broad tuning function. And this is noteworthy because it suggests that no one neuron encodes precisely the direction of movement. The responses, the contributions of any one neuron are simply too broadly tuned to achieve the functional goal. So how must this happen? Well, the functional goal is achieved through the encoding of direction in a population of neurons. So what was found as many neurons were recorded in the precentral gyrus during directional movement is that while no one neuron seemed to be sufficiently tuned to produce the movement, that a vector could be defined by the active population that aligned quite precisely with the movement that was actually performed. So for example, as the monkey moved the joystick up and to the right, we see this big red arrow that indicates the direction of the movement that was performed. There is a population of cells that are active, and if one then computes the vector average of their tuning functions during the execution of the movement, we find a vector average that aligns very nicely with the direction of movement. So for each movement that was executed, there's a population of neurons that are contributing to the directionality of the movement that's actually expressed. It's as if there is some kind of ensemble code for the direction of movement, with many neurons contributing information from which the precise movement is governed. So this then is an example of what we call a population code in the cerebral cortex. We find similar codes, we think, all over the cerebral mantle, with the idea being that for the most part, single neurons are not sufficiently tuned to express behavior, but rather the behavior is the product of an ensemble code reflecting the activities of an entire population of neurons. At this point, I think we're ready to wrap up this portion of the tutorial, and when we come back, we'll talk about the cortex just in front of the primary motor cortex, what we call the premotor cortex. Well, welcome back. I think we're ready to continue to discuss the organization of upper motor neurons and focus on the premotor cortex. So my learning objective for you is that I want you to be able to discuss the organization of the premotor cortex and its contributions to the control of volitional movement. So I'll remind you again that the premotor cortex is a region of the posterior frontal lobe that sits just in front of the precentral gyrus. It might include a bit of the anterior bank of the precentral gyrus, but it extends into the posterior part of the superior, middle, and inferior frontal gyri. And it also extends onto the midline of the hemisphere. Here, for example, is our paracentral lobule, so this is where we would find our primary motor cortex, and then just anterior to it, in the medial bank of the superior frontal sulcus, and then in the banks of the cingulate sulcus, including the cingulate gyrus itself, we find a medial extension of that premotor cortex. Illustrated here in this slide is the distribution of the premotor cortex in a rhesus macaque brain, where the details of organization are much more clear, at least at this stage in our understanding. So where we find this more orangish color, that's our premotor cortex. Now, there has been a fairly radical change in our thinking about the premotor cortex in recent years. Some time ago, we imagined some kind of hierarchical organization where the premotor cortex was primarily involved in planning movement, but the actual execution of the movement required signals that were derived from the primary motor cortex. So it was thought that the premotor cortex existed at a higher plane in some kind of hierarchy for motor control. Well, those concepts are beginning to unravel as we have a greater appreciation for what's actually encoded in the motor cortex. But we also understand much better now that the premotor cortex itself gives rise to descending projections to lower motor neurons. So the argument for premotor cortex being conceptualized as a higher plane for motor control seems to be losing some of its attraction. I think the concept that is becoming much more viable and frankly much more interesting and productive for understanding upper motor systems is the notion that this premotor cortex actually comprises a mosaic of areas that have some kind of modular organization, reflecting the encoding of ethologically important motor behaviors. Now, it is helpful to find some way to simplify our understanding of this mosaic, and I would suggest a means of simplifying would be to recognize a medial part of our premotor cortical mosaic and a lateral part. Where exactly one would make that division is not so clear, but perhaps one would roughly subdivide this premotor cortex at about the location of the superior frontal sulcus. So our medial division of the premotor cortex corresponds to what in the older literature was described as a supplementary motor area. So for those of you that are looking at other resources in support of your learning, you may run into that phrase, supplementary motor cortex. It certainly is still used in both clinical and scientific discussion. But for our purposes, we'll simply consider that supplementary motor cortex, or SMA for short, a division that is localized here to the medial part of the premotor cortex. Now, included in that medial part of the premotor cortex is a very interesting set of regions here in the banks of the cingulate sulcus that we'll spend just a little bit of time talking about. They seem to be involved in the expression of emotional behavior. Out on the dorsal lateral convexity of the hemisphere, we see other regions of this medial premotor cortex that are concerned with organizing bimanual activities. And this implies that they have a rich set of callosal connections as they do. So bimanual coordination seems to be a function of this premotor cortex. There's also a division of this medial premotor cortex that is especially concerned with governing voluntary saccadic eye movements. We call this region the frontal eye field. And this is a part of the premotor cortex that helps to orient our attention, and therefore what we're looking, across the midline to some location in the contralateral visual hemifield. Now, generally speaking, these medial parts of the premotor cortex seem to be especially involved with organizing movements that are self-initiated, movements that are not necessarily triggered or directed by sensory cues. One example of what I mean by this in the frontal eye field would be turning our gaze to some object in the contralateral part of the visual field that we intend to look at. Perhaps there's a clock over to your right and you want to look towards your right to see what time it is. So there wasn't necessarily an emergent stimulus that just grabbed our attention. Rather, there was an intentionality about the shift of gaze, and that intentionality was self-initiated. Now contrast that, if you will recall, to the role of the superior colliculus in organizing eye movements. If you're outside and a bolt of lightning happened to strike somewhere off in the distance to one side, you may very well make a reflexive movement of eyes and head posture in order to see that bolt of lightning while it persists. Well, that is a reflexive saccade that's coordinated at the subcortical level through circuitry that involves the retina sending signals to the superior colliculus, and then the colliculus sending output that governs the movements of the eyes and the muscles that orient our head and our neck. We call that a reflexive saccade. That wasn't self-initiated in the same sense as the desire to see what time it is. So now let's turn our attention to the lateral parts of the premotor cortex. Here what we find is a mosaic of areas that are involved in organizing movements that are often guided by sensory information, or at least with the interactions that we might have with the world around us. And that would include social interactions as well. For example, we find areas that exist here in the inferior part of this premotor cortex that are especially concerned with social communication. Now, for us in the human brain, we can recognize one region that is of great interest, and it's found in the posterior part of the inferior frontal gyrus. And this is a region that goes by the name Broca's area. So Broca's area is part of this premotor cortex that organizes the vocal motor apparatus for the production of speech. There are very likely nearby regions, also part of this premotor mosaic, that is involved in the production of language in written form, either through the act of writing with a writing implement, or of course now we can touch on keyboards and touch screens. And that is a motor act that is involved in communicating thought and semantic content through symbolic representation. And that seems to be one function of these more lateral and inferior divisions of this premotor mosaic. Also, in this same region of the premotor cortex, we have some really fascinating neurons that have captured worldwide attention in recent years. These are neurons that are concerned not just with the expression of movement that we make with our own body, but also encoding the intentions of movements that we see other individuals perform. And because of this mirror-like capacity of encoding our own movements and observed movements, such neurons with this behavior are called mirror motor neurons. Mirror motor neurons were discovered quite accidentally when recordings were being made in this lateral premotor cortex while monkeys were engaged in visually guided reaching behaviors to pick up a small object. And what was observed is that when the monkey simply sat passively while the experimenters reloaded the apparatus by executing a very similar type of hand movement, the neurons in the premotor cortex of the monkey's brain responded at the sight of the human hand performing the very similar act that the monkey himself was about to execute. This is illustrated here in this figure from our textbook. What we find is a recording from one such neuron in the lateral part of the premotor cortex, and we're looking at raster plots such as what we saw previously. So each row is an individual trial, and each little tick mark represents an action potential. What was really surprising at the time was that the very same neuron was seen to increase its firing rate when a very similar act was observed in a different agent, in this case, the hand of the experimenter. Well, as you might imagine, this was a really shocking discovery that neurons in the motor cortex might represent movement observed in another person. So quite a lot of work has gone on in the last 2 decades or so to really explore the encoding of movement intention that is observed here in this lateral premotor cortex. What was discovered is that these neurons seem to care about the agent itself. If a tool is used, for example, to perform a particular movement goal, then this neuron might not be concerned with that particular behavior. In this example, if a food pellet is retrieved with a pair of pliers, this neuron doesn't seem to fire, whereas the natural movement that the monkey makes or is observed by a human hand will elicit a robust set of discharges in this premotor neuron. Very interestingly, if the monkey is prevented from actually observing the contact of the hand with the food pellet, that is the consummation of the motor act, by occluding that view with some kind of a screen, we still see activation of this so-called mirror motor neuron in the lateral premotor cortex. It's as if what this neuron is encoding is the goal of the behavior or the intention of the behavior, which in this case is executing a precision grip to retrieve a pellet. Even when that act is not observed, there's still activity in this premotor neuron. So the discovery of these mirror motor neurons has really led to, I think, a really remarkable hypothesis, and that is that the lateral part of the premotor cortex is involved in encoding the intention of movement, and that movement intention might provide a foundation for social cognition, because these neurons are concerned not just with my intentions, but also with your intentions. When we observe the activities of others, we might actually represent that behavior within our motor system, and that might give us a sort of knowledge or understanding of the intention of that motor act. And this is a very active and frankly quite controversial domain of cognitive neuroscience at the moment, so we don't quite know how this is going to all play out. But I do suspect that this is an important discovery that is going to inform our understanding of social cognition, perhaps even the basis for disorders that lead to impairments in social cognition, such as some developmental disorders that result in the autism spectrum of disorders. This also provides some intriguing possibilities for intervention. If in such neurodevelopmental disorders of cognition there may be an underlying problem with the motor system, then perhaps there is a movement-based intervention that might help to restructure connections in this part of the brain that could produce some functional benefit to the patient. Well, that's total speculation on my part at this point, but I think it's an interesting idea that's worthy of some study. Welcome back to this tutorial on upper motor neuronal control of movement. Now I'd like to turn our attention away from the cortex and into the brainstem. My learning objective for you here is that I want you to be able to discuss the neural centers that give rise to medial descending projections from upper motor neurons in the brainstem to lower motor neurons associated with the ventral horn of the spinal cord and even in the brainstem itself, in the motor nuclei of the cranial nerves. So our brainstem upper motor neurons include primarily two sorts of populations of cells. There's a population of neurons associated with the vestibular nuclei, and we've talked some about these in a previous tutorial. And then there is a population of neurons that emanates from the central core region of the brainstem. That is the core of the tegmentum of the brainstem. We call this core the reticular formation, and the reticular formation really extends throughout the entire brainstem, but the portion of it that gives rise to projections to lower motor neurons is primarily found in the pons and in the medulla. Okay, so let me just take a minute or two to remind you of the projections that arise from the vestibular nuclei and descend and terminate in the spinal cord. You'll recall that there are vestibular spinal connections that are concerned with mediating reflexes that are sensed by our vestibular apparatus, and that sensory signal is going to be some kind of an acceleration or movement of the head. So this movement could be a rotational acceleration of the head, which would be sensed by our semicircular canals. It could be a linear acceleration or a tilt of the head sensed by the otolithic organs. And these signals might very well necessitate an adjustment of posture. And in postural adjustments imply that there must be some change in the output of the spinal cord involving the axial muscles, the proximal limb muscles, and perhaps even the distal muscles of the limbs. And these reflexes connecting head movement to postural adjustment are mediated via these vestibulospinal connections. So there is a lateral vestibulospinal tract, which arises from our lateral vestibular nucleus, one division of this vestibular nuclear complex. And this projection is primarily an ipsilateral projection that runs down the spinal cord and terminates among the cell groups that are concerned with extensor tone, primarily in the lower extremities. So one example I've given you before would be standing up on a bus or a train that is unexpectedly encountering bumps or jolts along the path. And so these are sensations that are being detected by the vestibular system. And in order to stay upright and not fall over as the floor is moving under your feet, there needs to be a moment-to-moment adjustment in posture. And that's what's mediated via this lateral vestibular spinal tract. Now, there are other divisions of this vestibular nuclear complex that give rise to a medial vestibular spinal tract. And this projection is much more bilateral than our lateral vestibular spinal tract, and it's terminating among medial lower motor neuronal columns. And these columns are mainly found in the cervical region of the spinal cord. So one example of the operation of this medial vestibular spinal tract would be the reflexes that are elicited with a sudden forward rotation of the head. So what I'm thinking of is tripping and falling. So in order to brace ourselves for the impact with the ground, we want to get our face out of the way, so we dorsiflex our neck, and we often extend our arms to brace ourselves against the fall. So that's a reflex. So the forward acceleration of the head triggers this reflexive response that's mediated via the cervical levels of the spinal cord. That's our medial vestibular spinal pathway. Now, I want to make just a couple of points and then we'll be ready to move on. I don't want you to be confused about the nomenclature of these vestibular spinal pathways, especially the lateral vestibular spinal pathway. This pathway is somewhat lateral in the anterior and medial white matter of the spinal cord, but we still consider it part of our descending medial systems for upper motor neuronal control of posture and the more proximal muscles of the limbs. So consider together these vestibular spinal projections part of that medial descending system that adjusts posture and sets the gain of local segmental reflexes. One additional point I would make about these vestibular spinal connections is that they are operating on sensory feedback. So there is some kind of action that's being sensed, and then an adjustment is made subsequent to that action. So this is a feedback operation, adjusting posture in response to a sensory signal. Okay, let's move on and talk about the reticulospinal system. So the reticular formation is really a complicated network of structures that we find throughout the tegmentum of the brainstem. So indeed, the reticular formation is a complicated network of neurons that spans the core of the brainstem. One can even argue that it's continuous with the intermediate gray matter of the spinal cord inferiorly, and continuous with the lateral portions of the hypothalamus on the superior side of the brainstem. Well, within this network of cells, we now know that there are actually discrete circuits, and many of these circuits have specific functions with respect to visceral motor control and somatic motor control. There are elements of the reticular formation that do in fact project broadly and diffusely throughout the nervous system, and those are projections that have an important role in modulating brain function in various regions. Broadly speaking, we recognize the rostral part of the reticular formation coming from the mesencephalon and from the rostral part of the pons as having those kinds of projections that modulate activity in the forebrain. The more inferior part of the reticular formation in the caudal pons and in the medulla, that's where we tend to find these circuits that have a kind of premotor function. That is, they help to coordinate the output of lower motor neurons. Many of these lower motor neurons are concerned with visceral motor functions, including cardiac rhythms and breathing rhythms and perhaps even digestive rhythms. Other networks are concerned with somatic motor function, and here's where we imagine our paramedian pontine reticular formation coming in and helping to organize horizontal gaze movements. But there are other circuits within this reticular formation that are concerned with modulating the gain of segmental reflexes in the spinal cord, as well as making adjustments of posture. So to go back to the previous slide for a moment, I would highlight that there are indeed projections from this reticular formation down through the anterior medial white matter of the spinal cord, and then near their level of termination, they tend to bifurcate and supply bilaterally the medial columns of lower motor neurons that are found in the spinal cord. Now, one key way to differentiate the contributions of the reticulospinal connections from those of the vestibulospinal connections has to do with time. You'll recall that I made the point that the vestibulospinal projections are operating as a feedback circuit, taking a sensory signal, the movement of the head, and implementing a motor adjustment. So in this sense, the vestibulospinal system is operating based on information acquired in the past. The reticulospinal system is concerned primarily with making anticipatory adjustments of posture. That is, making adjustments of posture that anticipate the actions that are about to happen. So here's an example that helps me understand the differentiation of these two systems. Imagine a sprinter, and that sprinter is in the blocks awaiting the sound of the starter's pistol. So that sprinter has just heard the words ready, set, and now there's an anticipation. There is a time of preparation of the body, especially the lower body, for this explosive motor action of sprinting out of the starter's blocks and engaging in this pattern of locomotion that is essential for competing in this activity, this sprint. That moment of ready and set is where our reticulospinal system will be engaged in setting the tone of the segmental circuits so that an explosive motor action can be executed with maximum energy and performance and skill. So this reticulospinal connection is going to be especially active during this time of ready and set. It will be preparing the circuitry of lower motor neurons for this explosive action that's about to happen. Okay, now you're that sprinter, the gun's gone off, you've exploded out of the blocks, and now you're running down the lane in the course of this race. Imagine that you stumble. That stumble is not something that you planned, and it's not something that you necessarily anticipated. It just happened. Well, that stumble is going to involve some unexpected acceleration of your head. That will engage the vestibular system, and hopefully you'll be able to catch yourself and maybe break stride a little bit, but you'll make some adjustment, I trust, and continue on in the race. Well, that stumble and the adjustment that allowed you to continue in the race is a function of our vestibulospinal system. The vestibular system sensed the unexpected acceleration of the head and an adjustment in posture and in the mechanics of locomotion were implemented via the vestibulospinal system. Well, I hope this image of starting and then running a race has been helpful and helps you to differentiate the contributions of the reticulospinal system from the vestibulospinal system. Well, if the reticulospinal system is going to serve to prepare you for action, to anticipate the activity that is being planned for, then it implies that it must know what that planned activity is all about. The implication anatomically is that there ought to be connections from our premotor cortex down to the reticular formation, and indeed there are. There are projections that emanate from our motor cortex that are informing these neurons in the reticular formation, probably bilaterally, about the activity that's about to be performed. And then the reticulospinal connections are those that actually make the appropriate adjustments of gain and muscle tone for the actions that are necessary. Now, there's one other structure that is often considered to be a collection of upper motor neurons in the brainstem that I want to mention. And it's found right here in the dorsal part of the midbrain. And what I'm attempting to identify for you here is the superior colliculus. Recall that the superior colliculus, it's one of the little hills that forms the tectum, or the roof of the midbrain, just above the cerebral aqueduct, which we see here. So there's the inferior colliculus, that's an auditory relay, and then the superior colliculus, which is the structure that I'd like to just mention very briefly here. So the superior colliculus is a motor structure that integrates sensory information from vision, from audition, from mechanosensation, and probably also from pain and temperature sensation. So these sensory signals are integrated in the networks of the superior colliculus, and a motor signal emanates out of the deeper layers of this structure. The superior colliculus is concerned with making reflexive adjustments of gaze and posture towards a sensory stimulus. So I already gave you the example of a bolt of lightning. Perhaps there could be an unexpected pinch of the skin, or maybe a sound that comes from some lateral position in our auditory environment that captures our attention. Either one of those three examples will often induce a reflexive shift of our gaze and our attention towards a particular stimulus. Well, how does the superior colliculus do this? Well, we've talked elsewhere about the role of the superior colliculus in coordinating saccadic eye movements through a portion of the reticular formation that coordinates movements in the horizontal plane or in the vertical plane. But there are also adjustments of the cervical spinal cord that are concerned with the turning of our head and the adjustments or the stabilization of our posture. Those adjustments that are mediated in the spinal cord are carried out by projections from the superior colliculus into the reticular formation, which then projects to the spinal cord. There may also be direct projections in the human nervous system from the superior colliculus to the lower motor circuits of the cervical cord themselves, but we know that at least there is a role for the reticular formation in mediating the signals that are coming out of the superior colliculus. So it's fair enough to include the superior colliculus in our conversation about upper motor neurons in the brainstem. Okay, we'll shift gears again and talk about the neural circuits for expressing emotional behavior. Welcome back. We now come to part 4 of our discussion of upper motor neuronal systems for motor control. And we come to a topic that has always intrigued me, and I've always found it very, very much fun to explore and to talk about. It's a discussion of what might be considered to be an emotional motor system. So my learning objective for you is to simply discuss the evidence that there actually exists an emotional motor system within our body's systems for governing movement. Well, I'm going to suggest again to you that one principal theme for motor control is that there are parallel pathways by which upper motor neurons can govern the activities of lower motor neurons. Now, we've spent a good bit of our time so far talking about what is depicted here on the left-hand side of this figure. That is a set of pathways that come from upper motor neurons in the motor cortex and in the brainstem that are concerned with the expression of skilled behavior, and this would involve the lateral pathways that come from the motor cortex to the lateral part of the ventral horn, being concerned with the fine control of the distal extremities. And then, in order to facilitate the expression of skill, there are pathways that come from the brainstem primarily that are concerned with adjusting posture and setting the stage for the expression of that skill. This involves making adjustments to the gain of our segmental reflexes. And so these brainstem systems send projections to lower motor neurons. There's in particular a role for the reticular formation of the brainstem in adjusting the gain of these pools of lower motor neurons. Now, what I'd like to turn to is the concept that in parallel, there are projections from other parts of the forebrain, other regions that give rise to connections primarily to the reticular formation of the brainstem, but perhaps also directly to pools of lower motor neurons in the brainstem and the spinal cord. And these regions of the forebrain that I have in mind are not in the posterior frontal lobe. So we don't conventionally consider them to be part of what is sometimes called the pyramidal motor system. That is, the parts of the motor cortex that give rise to the cortical spinal pathway that can be found in the medullary pyramid. For this reason, we call these extrapyramidal projections. And these extrapyramidal projections are coming from limbic centers in the ventral and medial parts of the forebrain, including some structures in the medial part of the temporal lobe, such as the amygdala, and parts of the ventral part of the forebrain, such as the hypothalamus and the diencephalon. So these so-called limbic centers then give rise to projections that run in parallel to those that are emanating from the posterior part of the frontal lobe and the upper motor neurons of the brainstem. So these pathways are largely involved in the expression of non-volitional behavior that we associate with emotion. These pathways are also those that are engaging our bodily systems, both our somatic motor systems and our visceral motor systems, for emergency situations that might require fight or flight. In order to try to be somewhat consistent with our lateral and medial scheme that we talked about for the control of volitional movement, we can likewise recognize a lateral and a medial scheme for the organization of these parallel descending projections in this emotional motor system. The lateral system is involved in expressing specific emotional behaviors, particular gestures of our facial region, for example, that communicate particular emotion, or particular postures that are involved in nonverbal communication. These tend to run through pathways that can be found in a more lateral region of the tegmentum of the brainstem. There are more medial pathways that are concerned with setting the stage, and these pathways are adjusting gain of reflexes, perhaps making it more or less likely that we might express a particular emotional behavior in our facial muscles. So let me give you an example of this emotional motor system in action. So I'm going to use some now quite old pictures of my son, who is now in his adolescent years. But in his younger years, it was very instructive for me to think about the operation of a pyramidal motor system for volitional control, and the operation of an emotional motor system, which moved the muscles of the face and our posture in a way that conveyed true emotional significance. Well, let me show you a picture of my son when he was about 18 months old. So typically when we would approach him, his name is Jonathan, and we would try to get him to smile. We'd say, smile for the camera, Jonathan. This is what we would get. Something that we sometimes call around here a cheesy smile, because sometimes people say cheese in front of a camera to try to create sort of a false impression of an actual emotional smile, when in fact it is a volitional motor act. And so this is what my son is doing here. So we're trying to get him to smile, and you can see that he is constricting his orbicularis oculi. He's trying to show his teeth here, and so in his 18-month-old mind, he is doing what we want him to do. He's expressing an emotion. But as parents, we know, well, this isn't really what we wanted to photograph. We wanted to really capture him expressing a true emotion. Well, if you knew my son, you know that he was a rather stoic young man and seldom truly expressed his emotion. This was a different setting where we put him up in a studio to get his picture taken, and the photographer is waving a stuffed animal in front of him, trying to entertain him, get him to smile. And so what we see here is about as close to a happy expression as we ever saw in our 18-month-old son. And contrast that to this very forced facial expression. It's quite easy to tell the difference between which expression came from a force of will and which perhaps reflects a true experience of emotion. So why do you think we're so good at telling the difference between a genuine emotional smile and a forced smile? Well, I think partly the answer is that we understand, whether we articulate in these terms or not, that there are different neural centers in the forebrain that are organizing the expressions in the human face that are conveying genuine emotion or a contrived volitional motor act that is meant to imitate that emotion. Well, the genuine emotion that we might experience and express in the muscles of our face is motivated primarily by the medial divisions of the premotor cortex that run into the banks of the cingulate sulcus. In fact, there's a region right in about this region of the human brain called the cingulate motor area. And this region is active when we express an emotional smile. However, if we present a cheesy smile, a pyramidal smile to a camera, that smile is being organized and executed by motor circuits in the lateral and inferior part of the premotor and primary motor cortex. This is a pathway that runs along the cortico-bulbar path in parallel with the cortical spinal fiber. So we consider this to be part of our pyramidal motor system. So quite different regions of the forebrain are responsible for the emotional smile and what we sometimes call the pyramidal smile. Now, if you view the tutorial that I've prepared to discuss the control of facial expression by upper motor neurons, you'll realize that there's also an important clinical sign that can be associated with the connection between these two different parts of the premotor cortex and the lower motor circuits of the facial motor nucleus. So please take some time and review that tutorial, because testing of facial motor performance is a key part of the neurological exam. I'd like to give you one more example of how our volitional motor systems and our emotional motor systems are integrated and coordinated to produce a desired motor output. In this case, the example is human speech. Now, my speech isn't particularly emotional at the moment. I suppose it could be, but you sort of know what you're getting from me at this point in the course. But nevertheless, we can break down speech into different components, right? So there is the pure active vocalization, that is just producing the sound itself, so that requires some coordination of motor output. And then there is, of course, what we're interested in, and that is the semantic content of speech that comes with the production of the phonemes that are representing the vowels and the syllables and the structure of the vocalization that conveys the semantic content. So it's necessary to integrate these two components of speech in order to actually produce understandable human language. So how does this happen? So these different aspects of speech are coordinated by motor output from the brainstem. So for example, one needs to coordinate the movements of the mouth, mouth opening, contractions of the floor of the mouth, the movements of the tongue, movements of the pharynx and the larynx, in order to coordinate that vocal motor apparatus for the production of the speech sounds that are necessary for intelligible language. But there's also coordination of respiratory rhythms and the muscles that force air through the larynx. So there's additional musculature that must be coordinated with the vocal motor apparatus found in the oral pharynx and in the larynx. And so this all occurs through the activity of lower motor circuits. And these motor circuits involve the nucleus ambiguus, the facial nucleus, the trigeminal motor nucleus, the hypoglossal nucleus, and the dorsal motor nucleus of vagus. So already you can understand the complexity of motor output that is required to make human speech. Well, when we back up now and consider how do upper motor neurons govern these lower motor neurons, we learn a little bit about the organization of parallel pathways. So concerning the semantic content of speech, this information is processed in our lateral premotor cortex in that very special part that we call Broca's area that is especially concerned with expressing human speech. Well, Broca's area, being part of our premotor cortex, sends projections to these lower motor neurons. It also sends connections through our primary motor cortex that interact with these lower motor circuits, and this helps give some structure to the vocal articulation of speech. But these signals would not be sufficient in and of themselves to produce speech. In addition to those projections that are emanating from the lateral part of the premotor cortex, we also need to engage our medial premotor regions, as well as structures including the amygdala and the hypothalamus that are going to be important for imbuing speech with emotion. And so there are relays from these ventral and medial regions of the forebrain to structures in the brainstem, including cell groups that are part of this periaqueductal gray region of the midbrain. And in turn, the periaqueductal gray sends connections to the reticular formation, especially parts of the medullary reticular formation, that then interact with our lower motor circuits. So the coordination of these two limbs is what is necessary to produce human speech. So of course, ultimately that coordination is derived from executive centers in the prefrontal cortex. Well, I think speech is perhaps a surprising example of convergence of parallel motor commands from very different parts of the brain that are responsible for creating the complete expression of human speech that has semantic content. We can think of that as a skilled articulation of our vocal motor apparatus, but also has the necessary gain, that has the necessary tone, and has structure that we interpret as emotion. And these signals are modulated by very different parts of the premotor cortex and even regions of the brain that are outside of our conventional collection of centers that we call the motor system. I'm thinking now of the hypothalamus and the amygdala that contribute to the expression of emotion in human speech. Okay, we've finally come to the final part of this rather lengthy tutorial on upper motor neurons, and what I'd like to talk about next in our final segment is the clinical picture that we would discover in an individual who has suffered injury or disease that affected upper motor neurons. Welcome to this tutorial on the neural control of facial expressions. I've got two learning objectives for you in this tutorial. I want you to be able to discuss the control of facial expression by upper motor neurons in the cerebral cortex. And then I want you to use that knowledge to predict patterns of facial weakness that might follow lesions of the cerebral cortex, the internal capsule, or the facial nerve itself. So, in order to begin this tutorial, I want to introduce a relatively common clinical phenomenon, and that is that a lesion involving the distribution of the middle cerebral artery, such as a stroke or a hemorrhage, often results in contralateral facial weakness. But that weakness is typically limited to just the lower part of the face. So the question is, why is this weakness restricted in this way to just the lower part of the facial muscles? And in order to explain this phenomenon, we need to learn something about how the muscles of facial expression are governed by different neurons in the cerebral cortex. So before we get there, let's just remind ourselves of some of the more peripheral components of this system. So let's consider a couple of study questions. First of all, what cranial nerve innervates the muscles of facial expression? So think for just a moment and record your responses. Secondly, where would you find the relevant motor nucleus? Now, if you checked your references, or hopefully you knew from your studies earlier, that the facial motor nucleus is what's responsible for supplying muscles of facial expression via cranial nerve 7, then you got the answer to the first one. But now what I want you to reinforce is, where in the brain would you find that facial motor nucleus? Is it in the thalamus, the midbrain, the pons, or the medulla oblongata? Now that we know that the facial motor nucleus is found in the pons, let's actually go find it in our digital atlas of the brainstem in Sylvius 4. So I've just opened our launcher window in Sylvius 4, and I will select from Brainstem Cross-Sectional Atlas all structures. And this will open up a collection of sections that are fully segmented and annotated that will allow us to find the facial motor nucleus. So now that you know where to look, it shouldn't be too difficult to actually find it. So first let's select a section through the pons, and I can do that simply by pulling down this slider in our navigational window to the left to about the region of the middle of the pons. And so now we have a section that is present, and I'll make it just a little bit larger for us. And as we mouse over structures, we see that these structures light up, as do labels. And I want to draw your attention to this central region of the brainstem. This is what we refer to as the tegmentum. It's the part of the brainstem between the ventricular system up above and the specialized white matter and gray matter structures that we find down below in various divisions of the brainstem. So that tegmentum is the core. That's where we find our cranial nerve nuclei. That's the part of the brainstem that's most like the spinal cord, where we have nuclei for sensation and for motor control. Now the facial nucleus, it's one of those motor nuclei that innervate muscles that have a particular embryological derivation. They come from the pharyngeal arches, and that's significant because this tells us where to look for the nucleus that grew out the axons that supply those muscles. And in this case, we're concerned with the muscles of facial expression, cranial nerve 7, the facial nerve, and the facial motor nucleus. Now these neurons in embryological development migrate to a somewhat lateral and intermediate position in the tegmentum of the brainstem. And for the facial motor nucleus, we find it right here. So now in yellow, we have our facial motor nucleus. Now one might think that these axons would simply take a straight shot out and exit the ventrolateral aspect of the junction between the pons and the medulla. But in fact, the course of these axons reflects this embryological migration of the motor neurons that I just mentioned. So this nucleus migrated from a more dorsal position, and as the neurons moved, they elongated their axons. So the axons from the facial motor nucleus actually wrap around and form this band of nerve root that wraps around the abducens nucleus. So the abducens nucleus makes a bit of a protrusion at the base of the fourth ventricle, and on top of that nucleus are these axons of the facial nerve that form a little bit of a hill on top of that protrusion. So we call this the facial colliculus. If we were looking down at the floor of the ventricle, we'd see these two little humps here, and that reflects the course of the facial nerve. And then from here, the facial nerve projects towards this angle here between the pons, the medulla just below, and then the cerebellum out to the lateral aspect. So there's the facial motor nucleus. This is the structure that is in question in this tutorial. So let's continue on now and talk more about the way that upper motor neurons provide input to this structure. Okay, well, I'm going to introduce the brain to you again using this brain model, and I want to draw your attention to that part of the motor cortex that is involved in governing facial expression. So what we're looking at here is the lateral surface of the right hemisphere, of course, and all of these red lines are meant to illustrate blood vessels that supply the surface of the brain. In fact, these would all be branches of the middle cerebral artery. Now, the motor cortex is found in the banks of the precentral gyrus, which is located right here. Of course, the central sulcus being this space just posterior to that precentral gyrus, which runs all the way from the longitudinal fissure in the dorsal midline to the lateral fissure, here between the frontal, parietal, and temporal lobes. Now, if you viewed my tutorial where I put my fingers on an actual human brain, you get the idea that the central sulcus is found roughly near the middle of the hemisphere. And as I'm putting my fingers down over the central sulcus now, my middle finger would be representing the precentral gyrus and my index finger representing the postcentral gyrus, with the space between my fingers representing the central sulcus. So that part of the motor cortex that governs our voluntary control of the facial muscles is found out here where my fingertip is located, in the inferior segment of the lateral precentral gyrus. So about the fingertip region of my middle finger is where we would find this relevant part of the motor cortex. So here we are, the inferior segment of the precentral gyrus. This is governing the movements of the contralateral face. Now, I need to be more specific than that in order to answer this clinical conundrum. What was discovered in recent years is that these upper motor neurons that are otherwise representing the face are actually governing the movements of just the lower part of the contralateral face. So as is illustrated in this brain model, this is the region of cortex that, when damaged by that middle cerebral artery infarction, is responsible for the weakness of the contralateral lower face. Now, what about the cortical representation of the upper part of the face? Well, we used to think that it was represented likewise here in the lateral part of the precentral gyrus. We now know from studies done in non-human primates that the control of the upper part of the face seems to be governed by a region along the mid-sagittal surface of the brain. So as I take this brain model apart, show you the medial face of the right hemisphere, I'll draw your attention to a region in the banks of the cingulate sulcus that's found right about here, just in front of our paracentral lobule. So in the banks of the cingulate sulcus is a division of the medial premotor cortex that we call the cingulate motor area. And it's this part of the premotor cortex that governs the movements of the upper part of the face. Now, one explanation to this clinical conundrum is that the upper part of the face is spared simply because the medial face of the hemisphere is not supplied by the middle cerebral artery at all. It's supplied by the anterior cerebral artery. But there's another anatomical fact that also works in favor of preservation of the upper part of the face, and that is that the cingulate motor area found here innervates the facial motor nucleus on both sides of the midline of the pons. So there's bilateral innervation from upper motor neurons here down to the relevant cranial nerve nuclei, the facial motor nucleus in this case. So let's look at a figure from our text from the box found in Chapter 17 of Neuroscience 5th edition, Box A. So this figure provides in schematic form what we've been talking about. So I'll first draw your attention to the representation of the face in this case, the right primary motor cortex. So we find upper motor neurons that send their axons through the white matter of the hemisphere, through the internal capsule between the thalamus and the basal ganglia, into the brainstem, through the cerebral peduncle, and into the pons. And as these axons enter the pons, they cross the midline and synapse preferentially on those lower motor neurons that govern the lower part of the face, as is illustrated here in this middle picture. So this explains the contralateral bias in the governance of these motor neurons that innervate the lower face. Now, I'll draw your attention next to the cingulate motor regions, which are found here along the midline of the hemisphere in the banks of the cingulate sulcus. These neurons project down to the more dorsal columns of motor neurons in the facial motor nucleus that innervate the upper part of the face. And the additional anatomical fact is depicted here in this figure, and that is that the facial neurons that govern the upper face receive bilateral input from the cingulate motor regions in each hemisphere. So this, together with the differential distribution of these upper motor neurons, explains why the upper part of the face is spared with the middle cerebral artery infarction. In fact, it's quite difficult to have a lesion in the brain that produces motor weakness in the upper part of the face because of this bilateral pattern of innervation. So even a unilateral stroke involving the anterior cerebral artery is likely to have spared motor control for the upper face. Now, also shown in this figure, just for completion, is what would happen with lesions that are deeper and more peripheral than a stroke involving the cortex. So notice, for example, the lesion illustrated by this letter B. This would be a stroke involving the white matter of the internal capsule. And such a stroke would affect the input from the precentral gyrus to the contralateral facial motor nucleus, replicating the impact of the stroke that one would see with a middle cerebral artery infarction out here to the cortex. There would be sparing of the upper motor neurons because of the bilateral innervation coming from the cingulate motor area. If one had a lesion to the facial nerve itself, or a pathological process, such as what happens in Bell's palsy, one would expect a distribution of weakness that would cover both the upper and the lower regions of the face. So in clinical practice, during a neurological exam, it's always important to test motor function individually for the lower part of the face and the upper part of the face. And this can be done by asking a patient to show their teeth, to check the lower muscle tone, and to have them raise their eyebrows, often against a little bit of manual resistance, so that the clinician can feel and see the strength of the muscles that are governing the upper part of the face. Now, there's some consequence for this anatomy with respect to how we understand the facial expressions that are so important to us as people. We read so much into the human face. We can interpret intention, we can appreciate emotion as we observe the movement of the facial muscles. And if you're like me, things get a little bit strange when you're sitting in front of a camera as I am now, especially if I'm in a portrait studio getting my picture taken by a photographer. We're often asked to smile in those circumstances, and somehow the smile typically is just not so authentic, is it? At least for myself, I have a terrible time in that circumstance trying to produce what looks like a compelling smile. What ends up is what looks like a fake smile or a contrived smile. And that's because that forced smile is really a product of our primary motor cortex out here in the precentral gyrus. The precentral gyrus is ultimately the motivator of volitional action. So that forced smile, it's really not reflecting genuine emotion often. It's something that we're trying to contrive for the moment. Now, should we express an actual emotion, hopefully it's much more authentic, communicates in a compelling way what's really going on in our internal state. That kind of smile is what we really want to capture in our photographs. That's the smile that's motivated by the cingulate motor area. So if you run across a photograph of an individual in a magazine or perhaps in your own photo album, you can tell very readily whether the smile is the authentic smile reflecting emotion or whether it's what's sometimes called the pyramidal smile, which arises from force of will from the premotor cortex, conveyed along a pathway parallel to the pyramidal fibers that ultimately control the movements that are expressed via our spinal cord. So, next time you see a person's face, think about these differential regions of the lateral precentral gyrus and the more medial cingulate motor area as they govern the integrated expression of emotion and other behaviors that we express with our face. Now, there are some important consequences for understanding the way these upper motor neurons govern the facial motor nucleus. There are folks that have strokes involving various pathways in the brain, of course, and some of them are known to affect either the voluntary expression of movement involving the face or the emotional expression of movement involving the face. So this schematic figure just illustrates what we've been talking about. There are parallel pathways coming from motor control centers in the brain that converge upon the facial motor nucleus and its neurons that ultimately activate the muscles of facial expression. We've described those neurons in the precentral gyrus that are associated with voluntary control over the facial motor nucleus. And if one should have a stroke involving those upper motor neurons or the pathway by which those neurons innervate the contralateral pons, one can have a condition known as voluntary facial paresis, in which there is weakness when you ask a patient to execute a voluntary movement involving the perioral muscles, such as showing their teeth. They may only do so on one side of the face. But if you can get that person to smile with the authentic smile of emotion, you may find a much more symmetrical response. And that's because the neural systems associated with the cingulate motor areas and their bilateral projections to the pons remain intact. Now, a much more rare condition can be found for individuals that have some damage, often requiring some bilateral injury to the cingulate motor area or their pathways that connect that part of the brain down to the pons. So damage to those pathways would produce the complementary deficit, something that we call emotional facial paresis. These would be an individual that, when told an amusing story, might only express emotion on one side of the face. However, if you ask those people to raise their eyebrows or show their teeth, they might do so symmetrically, because the pyramidal pathways from the lateral precentral gyrus are intact. Okay, there's one more point to consider before we conclude this tutorial, and that has to do with other cranial motor nuclei that you may be wondering about. So, what are the cranial motor nuclei that have a significant contralateral bias in the way upper motor neurons innervate them? And I'll give you a principle that will help you understand this. Essentially, whatever we can do with our cranial muscles that are lateralized, that implies a contralateral bias in the way upper motor neurons project to the relevant lower motor neurons. So some of us can wink with just one eye or not the other. Some of you are able to raise just one eyebrow. I can't really do that very well. I can only control my frontalis muscle, evidently through some symmetrical input. But if you could only raise one eyebrow, that would suggest that there is a contralateral bias to the innervation of those muscles. Okay, so some of you can raise an eyebrow, most of us can wink one eye or the other. We can often chew just on one side of our mouths, can we not? Imagine biting down on a hard nut. That's a lateralized activity. And finally, we can protrude our tongue just to one side of our mouth or the other. This is often something that's done in assessing the integrity of upper motor neuron pathways to the nucleus that controls the tongue. We might ask a patient to stick their tongue into just one cheek, something like that. And you can often put your hand on the cheek and feel the force of that protrusion. So these are all examples of lateralized motor functions that we can do with cranial muscles. And the relevant nuclei then would be the trigeminal motor nucleus that allows us to chew just on one side of the mouth, the facial motor nucleus, as we've been talking about in this tutorial, and the spinal accessory nucleus. Now this is the nucleus in the upper part of the cervical cord that allows us to shrug our shoulders, which we usually do symmetrically, but of course it is possible to fractionate that movement just to one side of the midline or the other. And then lastly, the hypoglossal nucleus, taking them in numerical order. This is the cranial nerve, nerve 12, that allows us to protrude our tongue. So these four nuclei are the cranial nerve nuclei that likely have a contralateral bias in the way upper motor neurons project down to the relevant lower motor neurons. The other motor nuclei are organized in a much more symmetrical bilateral fashion. For example, we don't cry out of one eye. We don't secrete saliva just from one gland by force of will. So there's coordination across the midline, either at the level of the cortex and or at the level of the brainstem itself. Well, I hope you've enjoyed this discussion of facial expression. We'll have much more to say about facial expressions when we talk about emotions later on in the course. But for now, I hope this has helped you understand the way upper motor neurons in the cerebral cortex govern the lower motor neurons that are found in the brainstem. Well, welcome back to our discussion of upper motor neuronal control systems, and I'd like now to turn to the clinical picture of what you will discover in an individual that has had damage to their upper motor neurons or the connections between upper and lower motor neurons. So my learning objective for you is that I want you to be able to discuss the signs associated with damage to upper motor neurons. Well, for those of you heading into the health professions, it will be critical for you to understand the difference between the signs and symptoms associated with lower motor neuron injury and those associated with upper motor neuron injury. And the problem here is time, and that is shortly after damage to upper motor neurons, the patient might present with signs and symptoms that resemble those associated with a lower motor neuron injury. What is observed with people in the first few hours to days, possibly as long as a week, following damage to upper motor neurons is a phenomenon called spinal shock. Spinal shock refers to an apparent failure of lower motor circuits to provide output to skeletal muscle. As a result, the patients are profoundly weak, the muscles appear to be flaccid, and the tone of the reflexes appears to be significantly reduced. These are all signs and symptoms that might be interpreted as a lower motor neuron injury. However, over time, as those first few hours to days to probably the second week after the injury, we see a quite different clinical picture. Those flaccid muscles have now become spastic. The low muscle tone has now increased significantly such that there's actually pathological tone, too much tone in those muscles. And those reflexes that soon after the injury were essentially flat now are actually hypertonic, that is, they're hyperreflexive. So let's compare our lower motor signs with our upper motor signs, assuming now that some time has passed and this initial period of spinal shock has abated. So what we would see is in the upper motor neuron syndrome, yes, we'd see weakness, which is also seen with lower motor neuron injury, but rather than seeing a decrease in reflexes, we would see spasticity. And spasticity involves an increase in muscle tone, an increase in deep tendon reflexes, and the phenomenon of clonus, which means that with a quick stretch of a joint, this is typically tested in the ankle joint, one might see a oscillatory movement or beating around that joint more than about 2 or 3 cycles. This is a sign associated with the spasticity of the muscles that flex and extend that joint. One might also see some special signs associated with damage to upper motor neurons. One such sign is called the Babinski sign. The Babinski sign is evident with the stroke of the sole of the foot with a sharp object. Now, for adults with an intact cortical spinal system, such a stroke from the heel of the foot towards the ball of the foot leads to flexion of the toes, or a downward turning of the toes. This is a normal plantar flexion response. Someone with damage to upper motor neurons might have a very different sign. With this heel to ball of the foot stroking with a sharp object, we would see an upward turn of the great toe and a fanning out or an extension of the remaining toes. So this is quite a different sign than what we observe with an intact nervous system. Now, the problem with the Babinski sign is that it's just not that good. That is, it has relatively low specificity and sensitivity. So a patient of yours may have an upper motor neuron injury, but may show a normal plantar response. So just be cautious in how you interpret a normal Babinski reflex. If you see an abnormal Babinski reflex, then you can have more confidence that indeed there may be damage to an upper motor neuronal system. So I want you to appreciate how the spasticity and the special signs contrast with what we see with damage to lower motor neuron injuries. So with lower motor neuron injury, we see decreased reflexes, we see decreased muscle tone, and over time, we'll see muscle atrophy. In the early stages of injury, we'll see this abnormal activity of muscle fibers in motor units that we call fasciculations and fibrillations. With upper motor neuron injuries, we tend not to see such a loss of muscle structure and integrity. So there may be some atrophy due to not much use of the affected body regions, but the atrophy will not be nearly as severe as what is seen over time with damage to lower motor neurons and the loss of innervation of that muscle tissue. So remember, with damage to upper motor neurons, the muscle is still innervated. The problem is that the output of lower motor neurons is no longer governed and regulated in a normal fashion. And that's why we see the emergence of these special signs. That's why we see the development of spasticity. It seems as though the aggregate effect of the descending projections, especially those that come from the reticulospinal connections, is to suppress the gain and the tone of our segmental reflexes. So once that overall suppressive influence is lost with damage, let's say, to the spinal cord, what we see below the level of injury is the emergence of higher gain in those segmental reflexes. And that's why there's increased muscle tone and hyperactive reflexes, and perhaps that's why clonus emerges with the rhythmical beating of the extensors and flexor muscles. Now, one final point I would make here about upper motor neuronal damage is that the kind of movement that is especially affected is the fine volitional movement that expresses skill. And I hope this makes sense to you. I think I've been trying to emphasize that much of the motor cortex and the cortical spinal system is concerned with the expression of skill with the distal extremities. So it would make some sense then, if there is damage to the motor cortex, that the greatest loss would be in the expression of skill with our hands, with our feet, with our lower oral facial structures. Now, if one had damage limited to the upper motor neuron systems of the brainstem, then we might expect there to be problems with postural control specifically. And if there were sparing of, let's say, the pyramidal system, one might expect there to be residual activities that could be performed with the distal extremities. So one can have a neuroanatomical framework for interpreting specifically the signs and symptoms of upper motor neuron injuries that are observed in any given patient. Well, I'm going to conclude this extended tutorial at this point about upper motor neurons. The one topic that's relevant that we have not yet discussed in this tutorial concerns the anatomy of the descending projections. And it's that topic that I'd like to turn to in our next tutorial. We'll have a chance to actually see the connections in cross-sections through the brainstem and identify the location of our critical pathways for upper motor neuronal control of lower motor activity. So I'll see you then, and look forward to having that anatomical discussion. Welcome to this tutorial on upper motor neuronal control. And what I really want to focus on in this session is the anatomical organization of the cortical spinal and cortical bulbar pathways. This topic relates to several of our core concepts in the field of neuroscience. I hope this subject will allow you to break down some of the complexity of the human brain. Nevertheless, I'm sure you appreciate by now that the brain is indeed the body's most complex organ. Now, the pathways we're going to be discussing today are genetically determined. They provide a foundation for upper motor neuronal control of lower motor circuits. So it's very important that we understand their organization and anatomical distribution. But that's not to say that even these pathways can't be changed by life experiences. Indeed, the strength of the connections between our upper motor neuronal pathways and lower motor circuits are subject to experience-dependent modification. And we think that this might be an important site of neuroplasticity as we acquire new motor skills. Indeed, many neural systems are changed with practice and with the acquisition of skill, but let's not forget that even these pathways that we're going to be talking about today may themselves be modified by experience. So I have several objectives for you in this session. I want you to be able to characterize the organization of the cortical spinal pathway from the cortex to lower motor circuits in the spinal cord. I want you to be able to do the same thing for the cortical bulbar pathway, only as we'll come to define this pathway terminates in the brainstem, not in the spinal cord. I want you to be able to recognize components of the cortical spinal and cortical bulbar pathways as we look at the brain from the outside, but also as we look at cross-sections through the brainstem and the spinal cord. And I even want you to be able to look into sections through the hemispheres and know where elements of these pathways will be located. Well, one way to help you consolidate the information that I hope for you to learn in this tutorial is to draw, to make a sketch. That sketch could be on a whiteboard, a chalkboard, a piece of paper. It could be made with a stick in the dirt at your feet. But what I would say about sketching is that the bigger, the better. So I want you to be able to sketch the cortical spinal and cortical bulbar pathways from cortex to lower motor circuits. And you might even want to include in your sketches the lower motor neurons themselves and their connection to the muscle fibers that they innervate. Okay, well, let me give you a broad overview of our pathways of upper motor neurons to lower motor neurons. And this is a figure that you've already seen in conjunction with our studies of upper motor neurons. And this pathway that you see here illustrates the way that circuits of upper motor neurons in the cortex and in the brainstem connect with lower motor circuits in the spinal cord. The main pathway that we're going to be concerned about today is the one that relates upper motor neurons in the posterior part of the frontal lobe, that is the motor cortex, to lower motor neurons that are found in the lateral part of the ventral horn of the spinal cord. Now, there are other kinds of motor neurons that are involved in governing the output of the spinal cord, namely three major sources of motor neurons in the brainstem. Probably most important would be the reticular formation of the brainstem. It supplies projections that terminate bilaterally in the medial parts of the ventral horn of the spinal cord. There's also descending projections from the vestibular nuclei of the brainstem, also distributed to the medial parts of the ventral horn of the spinal cord. And thirdly, there are projections from the superior colliculus to the upper part of the cervical cord, although in humans we think that this effect of the superior colliculus on the orientation movements of the head and the neck may actually be mediated through the reticulospinal connections. Well, so this is the broad view of our perspective of the distribution of upper motor neurons to lower motor neurons. And I think it's very helpful for you to keep in mind this medial to lateral somatotopy that we find in the spinal cord. So when we talk about our lateral cortical spinal fibers that are found here in the lateral white matter of the spinal cord, I want you to relate function to this anatomical location and appreciate that these axons are terminating among the lower motor neurons that are concerned with the skilled movements of the distal extremities. Meanwhile, those axons that we find in the anterior and medial part of the spinal cord are giving rise to projections that typically terminate bilaterally on the medial part of the ventral horn. So these would be terminations on columns of cells that are concerned more with posture and with the gross movements of the limbs via the actions of proximal limb muscles. So I want you to be able to associate these two different anatomical locations with two kinds of functions. The lateral column of the spinal cord is concerned with the expression of skill, whereas these medial systems are more concerned with the maintenance and adjustment of posture. Okay, well, let's talk now about our cortical spinal and cortical bulbar pathways. And to begin, we need to consider, once again, the organization of the motor cortex. So recall that the motor cortex is found in the posterior part of the frontal lobe, and we can divide it into a posterior primary motor cortex that occupies the anterior bank of the central sulcus in the precentral gyrus. And then just anterior to that primary motor cortex, we find the premotor cortex, which begins at the anterior part of the precentral gyrus and continues on into the posterior part of those three long parallel gyri that come to meet the precentral gyrus almost at a right angle, the superior, the middle, and the inferior frontal gyri. Okay, so that's what we have here in our premotor cortex. Well, both the primary motor cortex and the premotor cortex give rise to descending projections that contribute to the cortical spinal and cortical bulbar pathways. And to understand the difference, one needs to be reminded a bit of the somatotopy that we find in the precentral gyrus, and that extends into the premotor cortex as well. Now recall that the somatotopy in the motor cortex is a gross reflection of the somatotopy that we find in the somatic sensory cortex across the central sulcus and the postcentral gyrus. But the detail is quite different. Rather than there being a faithful representation in a point-by-point fashion of the contralateral body surface, what we see in the motor cortex is a map of movement intention. And that intention is roughly somatotopically organized in the following way, with movements that we intend to make involving our lower extremity being represented here in the paracentral lobule on the medial face of the hemisphere and just out a bit into the dorsal medial margin of the hemisphere. Well, as we progress in a lateral and inferior direction, we move from the lower extremity up through the trunk and into a large expansive region near the center of the motor cortex that is concerned with the movements that we intend to make with our arms and our hands. So there's a large upper extremity representation near the middle of the precentral gyrus and extending into the premotor cortex. While just inferior to that representation of the upper extremity, in this lower third approximately of the motor cortex, we find a large representation of the contralateral face, and specifically the lower part of the face. Now, I want you to learn more about the way the upper motor neurons of the cortex control the movements of the face in a separate tutorial that I've prepared called Upper Motor Neuronal Control of Facial Expressions. So I will refer you there for more detail about how the face is governed by not only this lateral inferior precentral gyrus, but also a fairly newly discovered region right about here in the banks of the cingulate sulcus that we call the cingulate motor area. And it will be important for you to understand how these two parts of our motor cortex relate to the divisions of the facial motor nucleus that govern the upper and lower parts of the face. So please see this other tutorial for a fuller explanation of this aspect of the connection between the motor cortex and the facial motor nuclei. Well, for the rest of our time, I'd like for us to focus then on the organization of these descending pathways, principally the cortical spinal pathway. I'll say a little bit about the cortical bulbar pathway, but I'll refer you mainly to this additional tutorial for some deeper explanation of the connections between cortex and those relevant cranial nerve motor nuclei. All right, so let's use this figure from our textbook, which I suspect is really quite small on the screen that you're viewing this on, but I'll try to use it nevertheless and refer you to the additional figure that we have in the tutorial notes. And then I want us to jump into Sylvius and really find the elements of this pathway in cross-section as they are displayed for you in the tutorial notes. Well, let's focus on the pathway shown in red. This is the cortical spinal pathway. So what we're illustrating, for example, will be the descending projections of a motor cortex neuron that might be found near the middle of the precentral gyrus. So we imagine this is a neuron that's concerned with the movements of the contralateral arm and hand. So this neuron grows an axon that runs through the white matter of the hemisphere, and it enters the posterior limb of the internal capsule. This is the means that allows this axon to pass through the hemisphere between the basal ganglia and the thalamus, and then eventually down into the brainstem. And these axons are a component of this massive stalk of white matter that we find on the ventrolateral surface of the midbrain called the cerebral peduncle. So these axons pass through the cerebral peduncle and they enter the pons. Now, along the way, these axons might sprout collaterals that supply additional structures that they may pass. For example, there may be a branch that supplies the red nucleus, which is a large nucleus of the midbrain. There may be axons that terminate among the pontine nuclei here at the base of the pons. There may be axons that pass through the core of the brainstem and terminate in the reticular formation, which is going to receive information from the motor cortex about the intention of movement. Well, in order to actually express the movement, these neurons have to make their way through the brainstem and into the spinal cord. And here's what happens as these axons pass through the medulla and enter the caudal part of the medulla where the spinal cord medullary junction is found. The vast majority of these axons will cross the midline in a structure that we call the pyramidal decussation. We can think of this maybe as a folding of the arms, where one medullary pyramid crosses over the other, and consequently, the bulk of the axons that were present in one medullary pyramid are now found in the opposite side of the white matter of the spinal cord. It's at that point that we call this the lateral cortical spinal tract. So roughly 90 to 95% of the axons that are present in one medullary pyramid will cross the midline and end up in the lateral column of white matter of the spinal cord on the opposite side of the midline. The remaining 5 to 10% of the axons or so remain on the same side of the spinal cord, and they are present in this anterior and medial white matter. From there, they tend to branch and give rise to bilateral connections to that medial part of the ventral horn that controls the more proximal muscles of the limbs. So this is a picture of our descending cortical spinal pathway. And I hope you're not too distracted by these collaterals to structures in the brainstem. This will serve the purpose of informing important circuits in the brainstem about the intention of movement. Okay, now let's turn our attention to the cortical bulbar pathway, which is shown here in this yellow colored axon. Now notice where this axon begins. It grows out of a neuron in the inferior and lateral part of the precentral gyrus. That's where we would expect representation of the facial region in our motor cortex. So this axon then runs through the white matter of the hemisphere, just as we saw for the cortical spinal tract axon, through the posterior limb of the internal capsule, into the brainstem where it runs through the cerebral peduncle, and perhaps along the way it too might give rise to branches that terminate in structures like the red nucleus. Now, as this axon passes into the brainstem, it will be directing its projections, its terminal fields, primarily towards one or another of the cranial nerve motor nuclei. Now, in this illustration, I think just for artistic simplicity, the branches are shown only to ipsilateral structures. But as is detailed both in the tutorial notes and in the separate video tutorial that I have for you on the control of the facial muscles, we know that these collaterals are actually bilaterally distributed. And for some structures, including the trigeminal motor nucleus, there may actually be a contralateral bias in that distribution. We know that's certainly true for the lower motor neurons that are governing the lower part of the face. That is, the lower motor neurons in the facial motor nucleus. But for the most part, we can think of these cortical bulbar pathways as bilateral. The only issue is the degree of contralateral bias. Okay, well, these axons are distributed only into the brainstem. They're cortical bulbar. This term bulbar refers to the brainstem, and many authors choose to restrict it even further to projections from cortex to the motor nuclei of the cranial nerves, not just all brainstem projections, but specifically those brainstem targets. I think we can compromise somewhat and consider the cortical bulbar pathway to be the means by which the motor cortex motivates the output of our cranial nerve motor nuclei. Well, just one additional quantitative point that might give you some perspective on the projections from the cortex to the brainstem. The dominant projection, really, from the cerebral cortex to the brainstem really is not cortical bulbar in the sense that I'm using this term here in this tutorial. They're actually cortical pontine fibers. So these are the projections that arise from all over the cerebral cortex of one hemisphere and terminate among the pontine nuclei that are present in the base of the pons. These cells in turn project across the midline into the cerebellar hemisphere, which is sitting out here somewhere. So if you review your understanding of the cerebellum and the cerebellar pathways, you'll recall this connection from cortex to base of the pons, and then from base of the pons to the contralateral cerebellum. This quantitatively is actually the largest component of axons in the cerebral peduncle. The minor contributions are actually the cortical spinal and the cortical bulbar pathway. Okay, well, let's return then to our focus on the anatomy of this cortical spinal and cortical bulbar pathway. And I'd like for us to look at some cross-sections in Sylvius and identify the locations of where we would find these axons if we were to look through histological sections of the brainstem and spinal cord. Well, here we are in Sylvius, and I've jumped into our photographic atlas looking at sectional anatomy, and we're looking at a section roughly through the middle of the brain in the coronal plane. And what I want to draw your attention to is the presence of the internal capsule. So there's a bit of white matter that's making its way through the middle of the hemisphere, separating the thalamus and the caudate nucleus on the medial side from the putamen and globus pallidus on the lateral side. And this provides a means for the axons of pyramidal cells that are sitting up here in the motor cortex to grow an axon and have it enter the middle of the white matter of the hemisphere that allows this axon to grow down into the brainstem and on through the brainstem. Okay, so there is the location of our internal capsule. So this is where we would find the axons of our cortical spinal and our cortical bulbar pathway. Okay, so let's section on through. And now we see the appearance of the midbrain. And we know that, of course, because we have this beautiful dark substance here in the midbrain. This is the substantia nigra pars compacta. The dark substance reflects the accumulation of a pigment called neuromelatonin by neurons that synthesize biogenic amino acids. And in this case, this is the location of our dopamine cells. So it's a convenient visual landmark that tells us we've entered the midbrain. Well, just inferior and lateral to our dark substance, our substantia nigra, we find the cerebral peduncle. So here's our cerebral peduncle. So now the axons of the cortical spinal and cortical bulbar tracts have made it down here into the cerebral peduncles. We can follow these cerebral peduncles even further into the brainstem, and we notice that now those peduncles have entered the pons. So we know that this is the pons because we have this broad base of the pons that's giving rise to these peduncles that are spreading out into the cerebellum. So these now are the cerebellar peduncles out in this direction. So our cortical spinal and cortical bulbar fibers are running down through the base of the pons in this location. Well, really to carry on this story, we need a closer look at the brainstem. So I'm going to leave this photographic atlas of the whole brain and enter our brainstem cross-sectional atlas. I think we can make this just a little bit larger. And what I'd like to do is to highlight again the location of these structures of interest. So we know that this is the cerebral peduncle, which is conveying the axons of the cortical spinal, cortical bulbar pathways. They tend to be found near the middle of the peduncle here, with the cortical spinal fibers being just a bit more lateral to the cortical bulbar fibers. And meanwhile, the rest of this cerebral peduncle is comprised of axons that are conveying signals from the cortex to the pons. I think we see this system even better in the next section. So now we have this nice, compact cerebral peduncle. Now, if you have your tutorial notes in front of you, you notice that I added a little bit of quantitative detail here. I said that in each cerebral peduncle, there's about 20 million axons, whereas in each medullary pyramid, there's only about 1 million axons. So these numbers give you a sense that the cortical spinal tract is really only a minor component of the overall output from the cerebral cortex that is distributed down through the hemispheres and into the brainstem and below. Really, most of this projection is going to influence the pons and its connection to the cerebellum. Well, we can follow this on through into the upper part of the pons. So here's the upper part of the pons, and notice what's happening. This cerebral peduncle is defasciculating, and so we can look for these axons here. These are our cortical bulbar, cortical spinal axons, but also included here would be some cortical pontine axons, and the defasciculation allows those cortical pontine axons to access these nuclei that are found here in the base of the pons. And those nuclei give rise to these pontocerebellar fibers, which provide input then out here to the middle cerebellar peduncle, which we'll see grow in dimensions as we get near the center of the pons. So here's our middle cerebellar peduncle receiving the input from these pontocerebellar fibers that are derived from the pontine nuclei. But notice that as we've passed from the upper part of the pons into now the middle pons, we're bringing back together again our compact cortical bulbar and cortical spinal fibers together with what cortical pontine fibers remain. And then as we pass on even further down into the pons, we will eventually lose most of the cortical pontine fibers, of course, because they're terminating there in the pons. And what we have left is going to be primarily cortical bulbar and cortical spinal fibers. Now, along this journey from cortex through the upper part of the brainstem, we've been giving off collaterals from the cortical bulbar pathway that will terminate among the appropriate motor nuclei. And, well, we have an important motor nucleus that's found right here in the caudal part of the pons. This is the facial motor nucleus. So we imagine that there have been some axons that are part of this cortical bulbar projection that are running through this basal region of the pons that are going to make their terminations here in the facial motor nucleus, specifically to the contralateral side. So this is one of those nuclei that have a contralateral bias, at least with respect to those lower motor neurons that are concerned with the lower part of the face. And again, I'll refer you to our tutorial on the governance of the facial muscles for more detail there. There's our facial motor nucleus. If we go down even further into the brainstem, we encounter another important motor nucleus of the brainstem, the hypoglossal nucleus. So now at this level, most of what we have for cortical bulbar and cortical spinal fibers is brought together in this nice compact bundle at the base of the medulla called the medullary pyramid. So for those axons that are destined to innervate the contralateral hypoglossal nucleus, we imagine that some of these axons in here might cross the midline and then make their terminations there in the contralateral hypoglossal nucleus. This is why, for example, we can direct our tongue out against one side of the cheek or the other because of this contralateral bias in our projections from the appropriate part of the precentral gyrus to this hypoglossal nucleus. Well, nevertheless, here we are in the medulla, and most of what we see here in the anterior midline of the medulla will be the cortical spinal fibers. We've passed through most of our cranial nerve motor nuclei targets, so the cortical bulbar pathway is nearly depleted, and that would certainly be so if we section down just a little bit further until we are clearly past our hypoglossal nucleus. The only cranial nerve motor nucleus left would be our spinal accessory nucleus. So with the exception of the projections to the spinal accessory nucleus, what we have in the medullary pyramid is almost entirely the cortical spinal fibers. Now, let's watch what happens to this medullary pyramid as we approach the junction with the spinal cord. What we see is that there's gradually a folding over of this pathway, like the folding of the arms, one on top of the other. So what's happening here is that there are axons that have been passing through the medullary pyramid on one side that are going to grow across the midline in this fashion. So they're actually going to course in a somewhat dorsal direction and thereby end up in the lateral column of white matter of the spinal cord. So once we get into the spinal cord, the axons that are present in one lateral cortical spinal tract are derived from the contralateral motor cortex. Okay, so here's one question that I'd like for you to consider, and I'll give you opportunity elsewhere to think about this again. Imagine I draw your attention to the axons that are present in, well, let's call this the left lateral cortical spinal tract. And I were to ask you a question like, well, where are the cell bodies that grew the axons in this left lateral cortical spinal tract? Okay, what would you say? Well, I would hope that you would understand this pathway that we've just talked through and recognize that these axons were grown by pyramidal neurons that have their home in the contralateral motor cortex. Now, we're talking about the lateral cortical spinal tract here, so one could even be more specific and say, well, in at least the center or the medial part of the motor cortex. Not the most lateral part, because that would be related to the brainstem and the cortical bulbar pathway. The cortical spinal pathway would be from the middle part of the motor cortex and then on medial through the paracentral lobule. Okay, so there's our lateral cortical spinal tract, and we can follow it all the way through the spinal cord, and it extends the entire length, although quantitatively the largest portion of the axons of the lateral cortical spinal tract are terminating in the cervical segments of the spinal cord, indicating a strong bias of this pathway to govern what we do with the distal upper extremities. Okay, now there remains one small component to consider, and this is something that we call the anterior or ventral cortical spinal tract. Let's just back up to the medullary pyramids for a moment. Now, here's our medullary pyramid, and as we've just described, most of the axons are going to cross over the midline and end up on the contralateral lateral column of white matter in the spinal cord, but not all. Some of the axons will remain, and they are just going to project straight on through this region of a decussation, and they will remain on the ipsilateral side of the spinal cord. So those axons might be present here, and then they will continue on down. And once we are into the spinal cord, they end up in a medial position on the anterior side of the spinal cord. And so these are axons that are derived from the ipsilateral motor cortex, and when they make their terminations as they pass on through the spinal cord, here we are down in the lumbar enlargement, these axons tend to project bilaterally to the medial columns of lower motor neurons that are found in the ventral horns. And this tells us that this anterior or ventral cortical spinal tract is concerned with what we do with our proximal limbs and our axial muscles. So it's concerned with posture and with just the gross movements of our arms and our legs. Well, I would like to conclude this tutorial at this point. I would encourage you to do this kind of exercise on your own, especially if you have Sylvius. Imagine what a cross-section through the cortical spinal or cortical bulbar fibers might look like, where they are to be found in that section, and then what structures surround them. And then you can begin to interact with your own sketches or perhaps your own models that you may wish to make. And imagine, okay, in my drawing, what would happen if we made a cross-section through, let's say, the midbrain? Where would we find our descending cortical spinal and cortical bulbar fibers? So this exercise of making visible your anatomical knowledge, I think, is quite valuable, because eventually I do want you to become familiar with the appearance of the human brain, not just in gross specimens as we see it from the outside, but also through histological sections, such as what we find here before us in our brainstem cross-sectional atlas. Okay, so the best thing you can do is to draw and to discuss and to join in the conversation that we have in our global learning community here in medical neuroscience. So be looking for a peer assessment that will encourage you to engage in just this very kind of activity. So you'll be hearing about that on the website. And otherwise, I hope you enjoy your studies of the cortical spinal and cortical bulbar pathway as you make visible your understanding of this important pathway in the human brain.